Who do we send the presentation to? Natalia?
Yes, please.
Okay. Let me see, I'll see if I make sure I have your email address.
Just in case, it's Natalia Perdomo _CFMC@yahoo.com.
Thank you.
And did you get Kate's presentation, Natalia?
No.
I will send her mine as well.
Hola,
Hello,
Kate.
good morning to all.
Thank you.
Wilson here.
Good morning, Wilson.
Ay, la de Jesús también. Hola, Jesús. Hola, Wilson.
If you can, please copy Leah Hi too. So we can
Buenos
go-
dias. Good morning, Natalia. Nicole Grio here.
Good morning.
Did you get my presentation?
Yes, Alida gave a pass to me.
Wonderful. Okay, thank you.
Thank you.
Good morning. Did you guys do roll call yet?
No, we have not started yet. Eddie, good morning.
Okay, morning. All right, thanks.
Natalia, you said Christina? Send it to Christina as well?
No, to Leah Hi
Julia, hi.
Rivera. Yeah.
Buenos días, Nati, ¿me oye?
Sí.
Sí, te oímos.
Ok.
Natalia, mira a ver si tú puedes hacer este presenter a Virginia.
Déjame ver.
Porque si puedes, entonces tú haces presenter a que le toque, que tú no tengas, lo haces presenter y le pides la... Y lo mismo le y cuando tú no estés, pues le hace lo mismo.
Es que no, porque voy al nombre y le doy a more y no me sale
lo
la
de
opción.
presenter.
No.
Estoy haciendo co-host por ahora porque hay un glitch ahí para que puedan presentar.
Pero Miguel, sin el beginner co-host pueden compartirlo.
Eso
Natalia,
es lo que
cambia.
pueden hacer, Graciela, por eso que te estoy diciendo. Pero no quiero mucha gente mezclando las cosas.
Estoy bregando con la página para que no, si alguien entra, no. Me voy a tardar un poco para que sepan, pero entiendo que las primeras presentaciones las van a dar los presenters, ¿no?
Sí, esas primeras tres, sí.
Ok, perfecto.
Gracias.
Miguel, ¿me escucha? Marcos.
Sí. Son las 9, ¿quieres dar 5 minutos
sí,
más?
Sí, yo voy a entrar ahora. Como
Pero
Natalia está
diga.
trabajando, Natalia está trabajando con el...
La persona no tiene que ver. Natalia puede seguir lo que está haciendo.
No, estoy pensando en el roll call como hacíamos siempre.
Por eso que si le das 5 minutos más como ayer, porque falta gente.
Ok.
Sí.
Lo hago
No, y
ahora.
también
Lo digas ahora.
Yo puedo hacer
Ok.
una pausa para el roll call y después seguir, no hay problema.
Perfecto.
Pero que
Voy-
más,
voy-
di que vas a dar 5 minutos más.
Good morning, everyone. December 9, 2020. This is the 172 CFMC virtual meeting. We're gonna wait 5 minutes more. We're gonna start at 9:05 a.m. Thank you.
¿A qué hora van a hacer break?
Depende de cómo sea la cosa.
Ok, perfecto.
I'm gonna see if the screen sharing works real quick, but then I can stop sharing if you want to put back up the welcome slide.
Go ahead and try it. There, we can see it.
Awesome.
Hola, saludo a todos y a todas. Buenos días. Es David.
Buen día, David.
Buenos días, David.
Buenos días a todos. Damaris Delgado por aquí.
Buen día, Damaris. Bienvenido.
Gracias.
Good morning, everyone. It's 9:05 a.m. We are ready to start the meeting. And today is December 9th, 9:05 a.m. This is the 172 virtual council meeting. We're going to start with the roll call. Natalia, can you help me?
Yes, good morning, everyone. I'm gonna start the roll call with Miguel Rolón, Graciela García Moliner, Díaz Rivera, Marcos Hanke, Virginia Chervet, Yamitza Rodríguez, Edian Ríos, Alida Ortiz, Angie Rizarri, Cristina Olán, Damaris Delgado, Daniel Olive, David Ortiz, Guillermo Cordero, Julia Magras, Jab McGovern, Janet Ramos, Jesús Rivera, Jocelyn D'Ambrosio, John Walter, Carlin Correa, Katie Siegfried, Laura Simo, Lauren Remsberg, Madeline Guyant, Manny Antonaras, María del Mar López, Michelle Shatter, Nelson Crespo, Nicole Grow, Nikita Charles, Noemí, Carlos Farchete, Richard Apperdern, Sarah Stevenson, Shannon Calai, Vanessa Ramirez, and Wilson Santiago. If I missed anybody, please identify yourself. I had an iPad here.
Edward
without
Schuster.
an- Thank you, Edward.
Miguel Borges, no.
Tony Blanchard, good morning.
Anybody else missing? Hearing none. We're gonna start with the first presentation. Today we have a list of very important presentations, very interesting, and let's try to make the best of our time. The first presentation is Deepwater Snapper on reef fishes in the US Caribbean, aging validation using bomb radiocarbon and preliminary longevity estimates by Virginia Chervet. Welcome, Virginia. Thank you very much. And go ahead.
Thank you for letting me talk about some of the work that we've been doing since about 2013 on reef fish and the deepwater snapper species. Can you hear me okay?
Yes, we do.
Okay, good. I thought I should check that. All right, so estimating ages and documenting longevity for fishery species is a fundamental step in our ability to sustainably manage fisheries. Information that we focus on doing research-wise is we do life history work, age, growth, and reproductive biology for Caribbean species. These data are then used by fishery scientists for some of the modeling work that they do as part of the assessment process. And in order to do those models, you've got to have at least you've got to have age data, and age data should be accurate. Otherwise, the models won't perform. They won't give you factual information, and that will make management difficult. So the tropical fisheries tropical fishery species that we work with are a little bit different from what a lot of other fishery biologists work with in the Southeast and the Gulf of Mexico. I've often actually heard multiple people from those areas that doing this kind of work isn't rocket science. So I think one of the reasons why they say that, and they think it's so easy, is because the species that they work with in those more temperate areas have really clear opaque zones on the otoliths that are easy to see, easy to count. That is not so for tropical fishes, though, the ones that we work with at least. Those residing along the shallower shelf habitats and the deep water slope species actually do not have otoliths with opaque zones that present super clear. So it's might not be rocket science working with fish aging, but it does take skill and experience. And it's naive of responsible scientists that oversee research projects that include fish aging as one of their study goals to assume that it's going to be easy to age their fish samples without the collaboration of experienced scientists. Many of the species that I'm going to talk about today have actually been inaccurately or incorrectly aged in past research. It really, it takes a lot of meticulous effort and a large amount of work to ensure that age data for these species are accurate and provide useful information for the populations. For those studies that have been published previously, obtaining and publishing inaccurate or incorrect age and growth data for fishery species can have detrimental consequences. So here's just some examples of the deep water species that we're currently working with, and just some of their otoliths. And these are all at the same scale, just to give you an idea. Winchmen have pretty big otoliths compared to queen snapper and vermilion snapper, for example. And all of them are really cool. It just takes a lot of work trying to figure out what we should be counting and just the methodology for aging our fishes. So that's why we have focused on validating age estimation for as many of the fish species as we can to ensure that we have an aging method that is actually providing the true age of our samples, of our fish samples. So we're doing this through application of the bomb radiocarbon chronometer. So that's C14. It was introduced into the atmosphere through nuclear bomb testing, starting in the 1950s up through until the 1970s. So basically this atmospheric C14 that wasn't there before got some really high concentrations relative to not being there before. It dissolves into ocean carbon dioxide, and then it gets incorporated into the aragonite skeletons, which is that calcium carbonate, biogenic calcium carbonate material that we see as part of the skeletons of hermatypic corals, which are shallow water corals, stony corals. Carbonate-based shells of things like mollusks, and then also aragonite or calcium carbonate-based structures of fishes. So the time-specific bomb radiocarbon aragonite record provides basically regional reference chronologies that we can then use to evaluate fish age estimates through comparing measurements from the core, the otolith core of a fish, or the islands core of a fish, which basically recorded the level of C14 that first year of life for that fish. We can take that and plot it against a regional record for our reference, and that'll give us an idea if we line up with the actual trend of the C14. So what I have here is I've plotted the coral record of C14 from Southwest Puerto Rico and from South Florida. And this is just to show you there's regional differences in this record. And it's a little bit different from when your objectives are looking at C14 in coral versus using it to age fish. And so what we actually had to do was to establish our own regional record to use for the validation purposes. And we were able to do that with using known age otoliths from red hind that were collected in the early 1980s all the way through 2020. And so we analyzed those red hind otoliths. We knew their ages. That made it so that we could plot what the C14 level was on our graph here versus the year that that otolith material was formed, or the birth year. And so all these X's are the ones that are the data that we've added to this record. And this is the tool that we now can use to validate age estimation methods for Caribbean species. So I'm going to give you some examples of some species that we've done this with. So this is blackfin snapper. This is an otolith section from a fish that was actually caught by the Department of Wildlife and Fisheries with BIDPNR as part of their deep water snapper collections, fishery independent collections. This actually ended up being the oldest blackfin snapper that's ever been analyzed, at least. The previous maximum age estimate for blackfin snapper came from a Florida fish, and that was 27. And so we've increased the longevity, the known longevity of this species to 45. This is basically the tool that we're using in order to validate our ages. And like I said, what we do is we take material from the otolith core that formed during that first year of life from the fish. We analyze that for the C14 level. Then we count all the increments on the otolith section. That gives us an estimated age. We take the date of collection, subtract that estimated age. So something for like this guy was caught in 2020, it was 35 years old. So it ended up having an estimated birth year of 1985. And so we plotted its C14 level right here. And then its estimated birth year of 1985, and it was right in the middle. And so we actually use statistical methods to analyze if our age estimate birth years are lining up with the reference series from the red hind. And so for all the species that I'm showing you today, with the analyses that we've done, we are statistically demonstrating that our ages our age estimates are accurate. And real quickly for blackfin snapper, some work was done for fish from Florida, North Carolina, and Georgia, and South Carolina, and they had a few Caribbean species. This was published a few years ago, and the growth curve that they got was very different from our growth curve that we've gotten from our preliminary samples right here. I've plotted the validated ages for our blackfin snapper versus the size, and then plotted calculated the growth curve. And so that's it with our observed data. And then this is just to compare it, just to show you that if we hadn't have done this research and validated our ages and knew that our ages were accurate, then when a stock assessment's done, they tend to use data, if we don't have it from local populations, data from other populations, like from Florida, will be used for the growth parameters. And so if that was done using the data from the study that was done a couple years ago by Burton et al., then that data would not be actually representing what we see in the US Caribbean. So again, it's important to do this work. This is just some other results for some other species that we're working with. I'll really just real quickly go through it. Kate is going to talk to you more about queen snapper and tell you about a little bit about some of the age work, but the maximum age for queen snappers that we validated so far is over 45 years old. We've actually only analyzed about 20. We've estimated age for about 20 cardinal snapper, and then of those we selected 5 to validate ages for, and the maximum age that we've gotten so far is 17 years, which they actually probably get a lot older. We just don't have a lot of samples to pull from yet. Another example of that is with black snapper. We've got 10 blackfin snapper samples so far. And of those 10, one of them had a age of 20 years. And again, that's a validated age. So with some of the species that we're looking at, their otoliths are teeny tiny. This is a coney otolith on a quarter, just to show you how small they are. These are the otolith sections for coney and Graysby. So we use the islands core instead, because the really small otoliths, you can't get enough material from the core to analyze, to do the analysis for the radiocarbon level. And so we actually use the islands, and this is showing you the islands. You can see these concentric circles. The island's core is what forms in that first year of life. So we extract that island's core, we analyze that for these species, and then for all the other species that I'm going to talk about after this, we use the island's core. Maximum age for coney so far for the Caribbean is about 30. Maximum age for Graysby is also up in the 30s. And those are validated ages. Another species that we've been working extensively with, and Jesus is going to tell you more about, is queen triggerfish. So previous work on queen triggerfish used the dorsal spine to estimate ages because the otoliths are teeny, teeny, tiny. There's an example right there. They're a weird shape and they're really hard to get out without breaking. So people were using the dorsal spine as an alternative aging structure. The maximum age that was documented for queen triggerfish in the US Caribbean was done by Manouche in the late 70s and 80s, early 80s, and he came up with a maximum age of 7 using the dorsal spine. We did the same thing and used dorsal spines and otoliths to estimate age. We plotted both results here on our radiocarbon chronometer. And as you can see, the dorsal spines do not appear to provide accurate age estimates. The otoliths do, and the maximum age that we've found so far is 21 years for this species. So that basically has tripled the longevity, our understanding of longevity in queen triggerfish. We're also investigating parrotfish species, 7 or 8 of the parrotfish species that occur in the US Caribbean. These guys also have teeny tiny otoliths. These are the Sparosoma species. And it was previously thought by Choate and Robertson that parrotfish in the US Caribbean do not live nearly as long as they do parrotfish. Don't live as long as in the Pacific. And so the previously estimated maximum ages, we've actually doubled or tripled depending on which one, but stoplight, you can see, has a maximum age that we found of 16 years, for example. Redtail, redband, yellowtail all have maximum ages that are much higher than was previously documented or thought. So for the scarce parrotfish species, so far we found a maximum age of 20 years, and that's for queen parrotfish. Striped parrotfish, we only have a few samples, and so they probably get much older than 7 years. We just need to continue to sample them and read otoliths and analyze a few more to get a better handle on their maximum age. And then we recently wrapped up a project with hogfish in collaboration with commercial fishers in the US Caribbean. And I just submitted the report for this. And this is just some of the results from it, just to kind of show you how we've applied this for a species. Maximum age that we found for hogfish is 20 years in the US Caribbean, and that was similar to what's been reported for Florida. And our biggest recommendation with the hogfish work is that we really need some fishery independent samples to further understand what's going on across the populations, just because we've got very different fishing methods among the islands for hogfish, for triggerfish, for all most species. So fishery dependent samples are great for getting some basic understanding of what's going on, but further research needs to be done for all the species with fishery independent samples as well. All right, that's all the species I'm going to tell you about right now. We're doing this for a ton more species though. We've got data for a ton more species. These are just some of them, including all the boxfish species, which are pretty awesome and unexpectedly get pretty old. And that's really all that I have for you today. thank you to all the fishers that have helped us. We couldn't do this without you. Thanks to the natural resource managers across the U.S. Caribbean. You've helped us tremendously as well. And yeah, thank you for listening to the talk.
Thank you, Virginia. Great presentation. I hope we have much more time to talk over and for questions. Let's open for 2 questions to follow up with the other presentations. Questions? Richard?
Yeah, good morning. That was really stunning, Virginia. Thank you very much for sharing all that. Can you put on one of your early slides that had the growth curve for the deepwater snapper?
Yes, sure. For the blackfin.
Yeah, I wasn't sure what species it was. So
I didn't write it down. Let me go back to it. There we go.
So my question is not your aging. I think that's really solid and incredibly significant. However, I'm looking at the growth curve and seeing how quickly it flattens, basically being driven by that one last point, where you have that other one that's way above it, the second to last point. And so I'm just questioning the variability on the growth curve that comes out of that, given that those 2 points are so variable, and I guess emphasizing the need to try to get a lot more of those bigger fish so you can really pin down what that L infinity value is.
Right. So this actually was research that we were just doing opportunistically. We were trying to get funding to actually do a full-blown study for age and growth of blackfin and some of the other species, but of deepwater snapper. But yeah, so this is just stuff that we pulled together ourselves, cobbled together money to purchase fish to look at this initially, so we'd have data. But yeah, you're right, completely. We don't have a lot of large samples. We need those larger samples in order to get a better understanding of what's going on in that region.
But to some extent, you know, documenting the age is perhaps even more important than documenting what the L infinity value is at that age. So I, again, congratulate you on these results. They're really going to make a difference in how we think about our fisheries. And I certainly hope you can use this to get the funding you need to do more.
Thank you.
Thank you, Richard. We have John Walter next.
Thank you, Chair. And Dr. Chervet, this is really groundbreaking work, and the center is really excited to see this work for a lot of these species because they've been super challenging for a long time. And specifically, we've got a queen triggerfish assessment coming up, and I think everyone involved with the fishery would really like to see the best data used. Do you think it'll be available early in 2021 for the upcoming assessment?
I think, and I don't know how politically savvy it is to say this, but so all of our triggerfish samples are fishery dependent, and there are major differences among the islands in the gears that are used. And so from our data, it looks like there's way different things going on with the populations, but it's probably just an artifact of gear selectivity. And so we've actually submitted a proposal to, I think it was SK. Last year we submitted a proposal to CRP, so we would get more data for fishery independent work on queen triggerfish, because that's what we need now. We need samples from across all 3 islands that are fishery independent, collected in a well-designed way in order to be able to really understand what's going on with the background populations. And so in saying all that, yes, we can have data ready from our fishery dependent samples, but the assessment will not be complete maybe without having fishery independent samples for age estimates at least.
Well, in actuality, we use fishery-dependent data all the time in our assessment models, and the models can handle that difference in selectivity between fishery-dependent and even fishery-independent data. It still comes from gear that has selectivity, if not availability. So
Just
we
wait
can
till,
deal,
yeah, wait till
and-
Hayes talks.
and it'd be great. The data scoping webinar is January 11th, and I really would invite your students and lab members and you to get involved in it, because I think it's going to really open the door for this assessment to getting the most recent and relevant information in.
Yeah, absolutely.
Okay,
All right.
thank you, Virginia. Thank you, Walter. We have one. I have Julian and Vanessa. Please be very brief for us I want to hear what, because Julia was involved in this project too. Very brief, your comments to move on, because we are a little late on the schedule. Go ahead, Julia.
Yes, good morning. Julia McGrath for the record. Excellent presentation, Virginia. I know we did a lot of work in trying to get this kicked off, and I know there's a lot more work to be done. And with that said, you know, I have the powers that be at the table or on the conference here today. And because we had some delays in getting funding, this goes out to Miguel and Marcos. If we put together a proposal, we are wondering if the council would support us with some funding to continue doing some assessments and collecting samples to keep this project moving forward, both for the deep water snappers and for the queen triggerfish. So I'm throwing that out there. I know there seems to be some extra money floating around because of not having the meetings the way we normally have them. And I would like to have some discussion, or you guys can have some discussion and being willing to help us to fund this project so we can continue. Thank you.
Thank you, Julian.
Thank you, Julian. Vanessa, very quick, please.
Yes, thank you, Virginia, for this presentation. I just want to comment that I hear that you need more samples about the triggerfish and hogfish. In the west side, we get a lot of hogfish weekly. So I just want to tell you, if you need more samples, just contact me and I'm gonna make that work for you. Thanks.
Absolutely, yeah, Wilson definitely is the one that helps us tremendously in coordinating getting samples. And so I'm sure all of us will be in contact soon.
Yeah. Thank you very much, Virginia. Next presentation. It's
Kate Overly.
Kate Overly, research on queen snapper. Go ahead.
All right, can everyone see my screen? Hopefully.
We can see it and we can hear you.
Fantastic. All right, so good morning, everyone. Thank you to the Caribbean Fishery Management Council for inviting me to talk about my research this morning. So my name's Kate Overly. I'm from the NOAA Panama City Lab in Florida. So today I'm going to talk about 3 projects fairly quickly that I'm leading in the Caribbean, specifically Puerto Rico, with a focus towards queen snapper, with the overarching idea of the development and implementation of a deepwater fishery independent survey that targets the deepwater snapper grouper complex. So the first project listed here is a habitat classification in Puerto Rico's deep drop fishery. Just below the title there are a few images taken from our deepwater videos. Those ones in particular range from depths from 250 to about 370 meters on the west coast. So this project actually originated as a pilot study which utilized a fishery independent hook and line survey designed by Steve Smith and Jerry Alt out of the University of Miami Erasmus. So we constructed a video camera system that was deployed on the commercial fishermen's deep drop fishing gear while they were fishing in order to evaluate the use of low-cost cameras to better inform the CFMC on fish habitat relationships going forward in the future. So the system worked great with the exception of the depth component. So we observed essentially that at depths greater than 250 meters, they really require some sort of auxiliary lighting. So ambient light was just not enough beyond 250. Given the focus of our project, they range down to 500 meters and greater, we've discovered. We ended up developing a 2-year project beginning in the fall of 2018 that utilized Jerry and Steve's fishery independent hook and line sampling frame and added the components of a deep water camera and LED light system in order to observe the benthic habitats and the fish communities. So to the right there, you'll see a diagram of the system we developed. Just in a nutshell, you have the example of the fisherman's vertical long line with hooks and a weight attached. And then what differs, obviously, is the camera system. So that camera system is attached to the fisherman's main line by 2 gangins. And then the system itself, the base of it is just constructed out of PVC starboard. That kind of cream colored rectangle on top there is just subsea foam. So that will keep the system pretty neutrally buoyant in the water. So it won't float too far up off the seafloor. It won't hopefully crash into the seafloor. And it keeps it at about a 45-degree angle, give or take. So that way we're getting a view of the habitat and the seafloor, but we're also seeing the fish and not just from a top-down angle. So that can be tricky to ID. So this developed system is deployable from center console vessels, tethered obviously to the commercial deep drop fishing gear. And we used it in the west, northwest, northeast, and southeast regions of Puerto Rico from a range of depths from 100 to 500 meters, just to cover that at the time known depth range of queen snapper and other species in the deepwater snapper grouper complex. So the overall objectives for our specific research questions was to describe habitat utilization of queen snapper using both the video and hook and line fishing data, in addition to generating length, weight, and collecting biological samples for age, growth, and reproductive studies on queen snapper and other target species as a collaboration with Dr. Virginia Shervette out of the University of South Carolina, Aiken. So our survey resulted in 471 videos documenting habitat and deepwater invertebrates over the course of 2 years. So in the bottom right there, you'll see some spatial coverage of our video and hook and line stations on that map. The red dots are our year 1 samples and our stations. The yellow are our year 2. So it gives you kind of an idea of where we were sampling. These are the first stationary videos that we know of utilizing local commercial fishermen in Puerto Rico to document habitat, fish diversity, and benthic invertebrates of these poorly studied deep water reefs. So we are currently finalizing our video analysis. So all videos are read for habitat classification, fish and invertebrate identification to their lowest taxon possible. We generate minimum counts, which is just the maximum number of fish observed in a single frame for each species, which obviously also results in the presence absence for each species. And then finally, we generate the percentage of bottom covered by specific biotic and abiotic features such as corals or rock using a computer software program. So this video data will be used to estimate factors affecting queen snapper distributions and abundance, in addition to generating data on species richness and a diversity index for the sampled sites. So the video data will also allow us to document patterns in the observed fish community, both on the hook and line and video. And just some examples of some of the video clips, those are just still images up at the top there, but you see some Wenchman snapper, you see some silk snappers, a sargassum triggerfish in that top left. So we see quite a range of fish from the deep water communities. So I mentioned that in addition to our video data, that we also generate catch data from our survey. So we generate fish ID and measurements, and that is recorded for everything that comes on board. And then we take the addition of sex and fin clips for deepwater shark species, and then weight and sex are also recorded for our target snapper species. So for our target snapper species, we have queen snapper, blackfin snapper, and black snapper. We collect biological samples, specifically otoliths and gonads, for all 3 of those whenever we catch them. So the biological samples collected, like I said, are being utilized in age and growth studies as a part of our collaborative work with Dr. Shervette and her team, which is essential to help us fill the large data gaps for the species in the deepwater snapper grouper complex. And then one of the most important aspects to our catch data is that all of it is co-registered with the multi-beam bathymetry data, so our mapping data, our video data, and therefore habitat, and of course we have depth and coordinates for all of our stations as well. So some preliminary results. This project was also funded by the Southeast Deep Coral Initiative in addition to the Cooperative Research Program. So we did have a focus on looking at habitat in deep water corals around Puerto Rico. So as this specific project is wrapping up and we're closing in on our analyses, our preliminary results identified 77 fish species on video and 22 different species caught on hook and line. In addition to that, we've identified over 100 invertebrate taxa, both sessile and mobile, throughout all 3 regions. So several locations on the West Coast exhibited high diversity of invertebrates, and we feel represent areas that may potentially be included in management decisions in the future. So our preliminary analysis for year 1 of fieldwork shows that the western region of Puerto Rico contained the most diverse and sponge communities in the largest numbers, with 15 different orders identified, which was followed closely by the Southeast with an N of 10, and lastly, the Northeast with an N of 5. And then just off to the right there, you'll see those are images from 4 West Coast sites, and those are just kind of a range of the corals we see at a variety of depths. And then if you look at the bottom left-hand side, you'll see a pie chart. Those are taxa documented in year 1, and it yielded a total of 1,200 individual coral and sponges, which made up a total of 6 classes. So we have a lot of diversity that we're seeing. So we are currently wrapping up the processing of our year 2 videos and are expecting preliminary results by the end of this month. So these data will be further explored to assist with describing habitat utilization and linkages between queen snapper and deep water coral communities around Puerto Rico, in addition to exploring habitat associations with other species as well. As I said, our focus is obviously on queen snapper for this project, but we do generate data on species such as silk snapper, blackfin snapper, any of the Priscipomoides genus, and so on and so forth. So into our second project that involves queen snapper. So as we wrap up our analyses for the original 2-year video project, we basically took what we learned to develop a more comprehensive U.S. Caribbean fishery independent survey utilizing stereo video and hook and line methods to assess the deep water snapper grouper complex in Puerto Rico, specifically the West Coast. So like I said, this project focuses on that development and implementation of a deep water fishery independent stereo video survey on the West Coast. And we are utilizing multi-beam mapping data for our survey design. And obviously we have this emphasis on targeting the deep water snapper grouper fisheries. And this survey, we did extend the maximum depth range. We've developed new gear that does go beyond 500 meters. So we extended it to 650 to try and get what we think might be the top end of some of these fishes' range, such as queen snapper, and try and incorporate that full depth. So the field component, which is currently slated to begin in January 2021, will consist of the deployment and the retrieval of a stationary baited stereo remote underwater video system. So for short, that's an SBRV. And that is attached to deep drop fishing gear, so can also be utilized off of commercial fishing vessels, and will record imagery of benthic habitats and fish assemblages where it is deployed. So that SBRV will have the capability of generating optical imagery using paired deep water stereo video cameras, which essentially allows for non-invasive methods to gather size composition and abundance data from both exploited and non-target species that are either not typically caught using the traditional hook and line methods, are wary of the fishing line, or are restricted, due to various fishing regulations. In addition to the stereo video, the system will also have wavelength modified LED lights. So instead of just having your white LEDs, we are actually utilizing red, orange, and amber. And that is to reduce the bias in video sampling for fish that are wary of lights at deeper depths. So for instance, queen snapper, cardinal snapper, and wenchman snapper do tend to kind of hang out on the outskirts of the light on all of our videos in our year 1, so we can still see them, but they don't come into the field of view. So we're trying to reduce that bias for this survey. So in addition to visual imagery, we are also deploying 2 fishing lines at each station in order to collect biological samples following the retrieval of that SBRV system. So these samples, again, will be going towards collaborative agent growth and reproductive studies with Dr. Shervette and her team at the university. And then by deploying 2 separate fishing lines, we're hoping to be able to test bait preference of species as well. So for instance, baiting with tuna on one line, baiting with loligo, squid on the other line, to see if there's any sort of difference in preference of snapper species, grouper species, and so on. And then to the right there, you'll just see an example of what our SBREV looks like, kind of just a diagram. The little red rectangles are the stereo cameras, and this is a side view just so you get the full idea of the system. But from the back, those are actually paired and set a distance apart so we can actually achieve stereo. So you can't see it in this photo, but there are 2 there for the paired system. So as for our expected outcomes, this project works to develop the necessary steps required to collect appropriate data to assess species in the deepwater snapper grouper complex, hopefully throughout the US Caribbean. So this will be achieved through the development and the build of this deepwater SBREV system combined with the wavelength modified LED lights, which, as I said, will provide a non-destructive method for measuring that fish length for species which avoid and are difficult to catch by hook and line methods. And then the survey itself will provide an unbiased georeferenced estimate of relative abundance for fish species and sizes of exploited and unexploited fishes for the west coast of Puerto Rico. And then overall, this project provides technology that will be directly transferable to other CFIS regions, where it can be reproduced at low costs to gather data on distribution, abundance, length composition, and so on for species of interest. And this can also be given to other organizations. Other organizations can definitely use this technology as well. And then lastly, onto our third and last project I'll be discussing with you all. It's age and growth of queen snapper. Virginia went over a little bit of this, thankfully, so you have kind of the background of the bomb radiocarbon chronometer and just the age and growth for the species in general. But pictured here is a queen snapper otolith for a fish with a fork length of 708 millimeters. And you can kind of see it just by looking at it, that the opaque zones are very difficult to discern. And you can kind of get an idea of how difficult the species can be to age. So essentially, the objectives for this project were to age archived and contemporary otoliths from the US Caribbean and the US Gulf of Mexico. So we're able to do a comparison between the 2 regions to see if there's any growth differences. So we have a lot of otoliths at our lab in Panama City. We have a lot of archived queen snapper otoliths. So we basically took a subsample across 3 decades, ranging from 1991 to 2019. Took a subsample of 300. And then in the US Caribbean, we have over 800 otoliths that come from varying sources, from the Puerto Rico DNER, commercial fishermen, Dr. Shervette and her team, and then obviously our project, Steve Smith and Jerry Alt's projects. We have a lot of sources giving otoliths to and contributing to this project. And those range in date from about 2005 to 2020, with a few gaps in between there in the early years. So given the difficulty sectioning and aging queen snapper, we are validating the accuracy of our age estimation via application of bomb radiocarbon chronometer using both otolith and eye-lens cores, which Virginia went over a little bit in her presentation. So the bomb radiocarbon chronometer, we're essentially using it because the aging error in tropical as well as deep water species can be exacerbated due to that environmental constancy that provides very little seasonal variability to drive that distinct opaque zone formation that makes it easy to count opaque zones. So queen snapper are absolutely no exception to this and are very difficult to age as a result. It took us a while to nail down a sectioning protocol for the thickness of the otoliths and so on. So because of this, we determined that age validation would need to be employed to accurately age this species and therefore prevent aging error and validate accuracy of our age estimation. So the pictures on the right-hand side here, real quick, we have our isomet saw. That is what we use to section the embedded otoliths. We take 3 sections of each queen snapper otolith. The top image there just shows how small and fragile these otoliths are in comparison to a penny. So those are queen snapper otoliths. And then the bottom 2 images there are eyes from queen snapper. So the one on the left, that larger eye, came from the fish with a fork length of 708 millimeters, so much larger. The one on the right came from a fish with a fork length of 178 millimeters. So our smallest sample, actually, very small fish, and you can see the difference in eye size there. And then the image at the bottom there is just an eye lens extracted out of the eyeball, and it's just in a gloved hand. So we actually peel the layers back and get to the eye lens core, and that is what we use in our validation. So our expected outcomes. So we are wrapping this up as well right now and hoping to get out a few publications to disseminate our results. But essentially, this project, age growth and mortality parameters are essential. The essential first steps to being able to compute that quantitative assessment on queen snapper in the US Caribbean, right? So this project is providing critical information on queen snapper life history, including validated age composition estimates for those US Caribbean queen snapper. We actually have a total of about 21 eye lens cores and 5 otolith cores from queen snapper that we used in our validation. Size distributions for the US Caribbean and the Gulf of Mexico, so we can do comparisons between the two. And then something that was very exciting was extending the longevity estimates for queen snapper. So our previous maximum age in the last CDAR that was conducted on queen snapper was 8 years old. So since then, we, with our results, our preliminary results that we've received, our youngest fish and our smallest sample at a fork length of 178 millimeters has been validated at an age of 5. And one of our largest at a fork length of 708 millimeters, so those two eyes on the last page belong to these two fish, that has been validated to an age of 46 years old, which absolutely dramatically increases our estimates of longevity for this species. And you can see those results plotted on the graph to the right as part of that linear decline with the coral and known age otoliths. And that's just, you can also see the red circles are all the eye lens samples that we sent in for validation, so you can get an idea of the spread of our data. In addition to all of that, we are also computing growth functions and estimates of natural mortality for queen snapper. And really quick, I just wanted to acknowledge a few of the collaborators that I've been fortunate enough to work with, and also I'm going to see if this video plays so we can do that while I'm talking. So there's quite a bit of distance, obviously, between Florida and Puerto Rico. So there are a lot of folks that have helped to ensure the success of this project. My direct collaborators, Andy David, Steve Smith, and Ryan Cayuette with NOAA Fisheries have assisted with the video work and the sampling frame of this survey, in addition to Dr. Shervette of the University of South Carolina, Aiken, who has contributed funding and a bunch of assistance to actually process all of our age validation samples, and has been invaluable to the Age and Growth Project. And of course, last but absolutely not least, the commercial fishermen, all of our observers, and the team at HJR Reefscaping, who is our contracting company on the ground in Puerto Rico, specifically Michelle Scher and Hector Ruiz. Without that field team on the ground in Puerto Rico, we would not have been able to complete this field sampling and data collection. So a huge thank you to everyone there. And that's just my email address at the top there. So if anyone has questions that they don't want to ask now or think of later, feel free to reach out to me at that email. And I think that's all I have.
Thank you, Kate. Great presentation. We are really behind the schedule. Let's have one question for Kate or two, and move along, please. Please send the questions to her email like she posted, and we can follow up on that. I wish we have a little more time. I have Stacy.
Stacy, you're muted.
Okay, can you hear me now? Sorry about that.
Yeah.
Okay,
Yes.
thank you. Kate, great presentation. I just have a couple questions. How many habitats did you identify during your study, and did you see any patterns in the habitat preference of queen snappers between like sex or size composition in your study?
So we have video for 471 stations. So all of those have varying degrees of habitat differences. So I can absolutely chat with you more about that. We basically classified our habitat according to the CMEX standard, for the Coastal and Marine Ecological Classification Standard. So I have all of that data in general classification scheme. But since we sampled so many videos, it probably wouldn't be great for me to sit and list them all out right now. But yeah, in addition to that, so we're also doing our analyses right now. So I don't want to say too much. There's a lot on kind of that first slide that I showed with that kind of rocky sponge habitat. We see a lot on that lower relief rocky sponge. But we also see, especially in the Northeast, a lot around sand. I don't know if that's due to foraging and that sort of thing, which it probably is. So we're going to try and be digging into that once we finish our basically our habitat classification and our percent bottom coverage for year 2. We're just about done with that, so we should have results for all of that very, very soon.
Thank you, Kate. We're gonna pass to the next presentation because we are very much behind the schedule. We need to gain some time. We have the queen triggerfish reproductive biology in the U.S. Caribbean by Jesus Rivera.
Marcos?
Sí, go ahead, Miguel.
Okay. We have a person that had a presentation at 10:15. So what we propose to do, I just talked to Graciela, is that right after Jesus Rivera presentation at 10:15, and we will play with the time. Right now it's about 10 o'clock. So at 10:15, we will have the presentation of coral reef, and then Alida Ortiz will follow with her presentation at 10:30, or right after the presentation of coral.
Okay, go. Let's start with Jesus. Jesus, welcome. Go ahead.
Hello.
Saludos, Jesús. Go.
¿Tú vas a presentar o tiene Natalia tu presentación?
Do you see the screen?
Not yours.
No, we're still seeing the Deepwater.
Okay.
So, Kate, stop sharing. There you go. Okay. Is that you? Okay. There we go. You have it. Thanks.
I have a question. They are seeing the full.
Yeah, we can see your notes.
Yeah, I mean, so
Change the display settings.
Now?
Go to the left, it says display settings.
Okay.
Where you've got your mouse on in slideshow. Just scooch over to the left a little bit.
Yeah.
Here?
Just go to screen setting and just have the slide projection only.
Okay, sorry.
No.
I'm new in this. I have a technical problem in this.
We can see it anyway. You can just go ahead and continue the way you have it.
So I don't understand. I have to do. Sorry for that.
Bueno, just use the presentation
Just
as
move
is.
the presentation as is.
Jesús, puedes cambiarlo en el presenter view que te dice display setting in the top. Tienes que cambiarlo en el presenter view.
Jesus, we can see your presentation well. If you don't mind, please, you can present like this. We can see very well.
Well, here, yeah, I think I got it.
Just go ahead and move the slides just the way you have it now.
Now do you see it?
Yeah, just have to now put it in slideshow mode.
You see it now?
Yes.
Okay, cool. My name is Jesus Rivera. I'm gonna talk about my presentation of reproductive biology of queen triggerfish in the U.S. Caribbean water, specifically in St. Thomas. I forgot that to add to the title, in collaboration with Dr. Sherbet and Julian McGrath. Again, effective fisheries management requires a detailed understanding of the life history strategies of management species. Queen triggerfish support productive fisheries in the U.S. Caribbean waters, but the lack of current species-specific life history information in the 2013 CEDAR, they cannot construct projection for the future stock. So we are trying to address the reproductive biology of the fish and also age. We already, Dr. Sherbet, talked about a little bit about what is our age work going. So some previous studies related to reproductive biology of queen triggerfish are data from Jamaica, from Aiken in 1983, Puerto Rico and St. Thomas for Manook and Drennan in 1987, Puerto Rico and St. Croix, that was part of my thesis in 2018, and in St. Croix with Brian et al. In 2019, the home range publication. Also, we have Brazil with Ferreira de Menezes in 1979. For Aiken, they just found in Jamaica that the queen triggerfish season go to January through March, then May, and then July to December. Manook and Drennan also show they have some problem in the collection of spawning capable fish. We're gonna look that in a bit. And they also report 7 years. You already Dr. Sherbet talked about it. And we have more publication on triggerfish, but they are not related to reproductive biology, so I didn't include it. But mostly are for diet stuff. Queen triggerfish, you have a picture from Dr. Nimet in St. Croix that we have a triggerfish in their habitat, but also in this picture we have something that looks that is the nest of the queen triggerfish. That is kind of similar to other investigators look in other triggerfish species like a great triggerfish, and they document well the nest and how they just take care of the nest, both of the sexes, and that is just something that we don't address because we don't propose to do that in our grant, but we also but we try to get samples and kind of area where the fishermen say, oh, we see a nest, but we don't have any report. Well, fishermen say that they see something similar, but we don't have any kind of specific data of where's the nest or a spawning ground in this particular case. The other 2019 work on St. Croix, they have presumptive areas of nesting, but it is in the closest area. And yeah, they're try and this picture just show how they try to clean the nest, and then the females came and select if they're gonna have to, if they just want to mate or not. So our study objective are determine and compare size, structure, and sex ratios. Determine size at sexual maturity if we can. Determine spawning seasonality. Our methods is just really, we have the 3 islands of the US Caribbean, that is Puerto Rico, St. Thomas, St. Croix. Something really important that you have to understand is that the 3 islands have they use different gears. Puerto Rico is the one that has the most gears for catch triggerfish. There are multi-species, so one gear can catch more than one species. St. Thomas and Croix are different, and they're gonna show a table later. And again, preference in the island for the fish are different. Also, they are close, but they are really different in what the people like to get from the fisheries, at least in terms of fish or free fish. So what we do, we just propose to target 15 samples per month and trying to cover all the size class per month. So we just get the fisherman, the information, the gear that they use. We measure the fish, we weigh the fish, and then we just proceed to remove the gonad. Also the age structure, and other stuff that are not related to this presentation. So we assess the sex by histology. Not all the people do histology. Most of our work don't do histology. You just open the fish and see if it's male or female, and that's it. So quickly, because we are running on schedule, that is the process. We get the fish, we extract the gonad. We use 3 different fixatives depending of the situation, and then we just a standard embedded and stained with hematoxylin and eosin. And then the reproductive criteria that we use is in that way for both sexes: immature, developing fish, spawning capable. That is where we just see whether the fish is spawning or not. They have a subclassification that is actively spawning that we can talk later if you want. Then regressing is after they spawn, regenerating, just preparing again to go to developing, and it's like a cycle. So you don't go back to immature and regenerating, you just go back to develop and continue the loop. Our results, I will be presenting the summary of sample collection. In the top in red is St. Thomas. That is the purpose of this presentation. The other 2 in orange are the Puerto Rico and San Croix data that are already published. So we have 690 samples in total. 7 are only fishery independent, and then 683 just fishery dependent. That is just has Dr. Sherbet explained that is a problem for some of the analysis, but also hearing the explanation of the guy that made the question. So we probably need more data because that is a data limited species. So we still need more data, and we'll explain why in a bit. So also a more specific result of fish collection. We have the depth range. They are vary a lot. We have the total number of fish that we already see it, the percentage of male and females per samples, per island, and then the mean size overall, male, female, and the unknown fish that is just fish that are just gutted, or they are just small, or we just miss the slide, and we just try to figure out and do it again. So, but are pretty low. We don't left sample behind. So we are continuously looking for it. Now I am showing in the left side, just show the distribution of combined sets for the 3 islands, just to an overview. Red is Puerto Rico, green is St. Thomas, that is the purpose of this presentation, and blue is St. Croix. Then in the right, you will see the weight versus length plots for males and females of queen triggerfish in St. Thomas. So I think that you don't see it so well, the equation, but the equation look kind of similar to the one that we just published through the other islands. So maybe it's good data. For size, structure, and sex ratios, all 3 islands, the size frequency distribution of male and female were significantly different, meaning that male are larger than females in the distribution. For sex ratio, the chi-square analysis showed that Puerto Rico was the only that different from 1-to-1 expected ratio, and St. Thomas and St. Croix, the sex ratio was expected. So there's no difference in the distribution of male and females in St. Croix and St. Thomas, but was different in Puerto Rico, having more males than female, I think, if I remember well. And again, like I was telling you, that is the length frequency analysis from St. Thomas. The other 2 are published already. And yeah, we can see in the site class 350 that then the males that are in blue, they start getting in a bigger proportion, and then just females start to decay with length, the frequency. So most of the question that we have from fishermen is what we do with the fish. So what do? We have a queen triggerfish gonad, male gonad. That is the gonad. Really weird to find that big gonad in triggerfish. And this is the accessory that they just put the sperm there and reserve the sperm there, and they probably use it when they're gonna use it when they're spawning. And in this side we have the histology of that. That will be the gonad, and that will be the accessory of the queen triggerfish. That is from an immature fish. And now we have in the left, we have the accessory, the purple, the deep purple or blue, depending how do you see it, is the sperm. And in the right side of the right picture, sorry, is the gonad. That will be the gonad in the cross section or transverse section. And this will be the central, I don't remember the name, sorry, I'm nervous. That they just put all the sperm go here to this tube and then just go to the accessory. The reserve for seasonality for the males of queen triggerfish for Puerto Rico and St. Thomas we are focusing in the red color, that is spawning capable criteria. They just show that we have spawning capable males around the year. So nothing different. We are not surprised with that. We see that with the males in most of the species are in that kind of way. But for this current study, for this current presentation, that is St. Thomas that we visited, we are looking also the red one. Also, it's again all year. We have spawning capable males around the year. So that will not help us to see if there is any seasonality on the species. So we was gonna look the female. That will a little bit next. And also in the right side of the screen, I will show you the size and maturity for males. The presumptive L50, that is where the fish. There is the point where the length where the fish are at least 50% of the fish that will be that length will be mature. So it's a 50% chance of it's mature or immature. For this particular case, 156. And then I just put a circle in an outside their value that was looking with Dr. Sherbet, but because I don't have access to the campus, we cannot. Let me just. The problem with that value is that we have only one immature male, and that is that value. And the analysis, the confidence interval for the L50 doesn't work. Was really weird because we have a lot of mature male before that value of immature. So we are comparing one immature against 340 something mature fish species, and then the size overlap. But I will show you in the resume later. Now we go with the female. You see the same. The female are more interesting. We have a big gonad for a queen triggerfish in the upper picture. In the picture below is I can say developing a female just by experience, but I don't have any idea. I mean, it's really hard to assess macroscopically a gonad for a fish. And then a representation how we look at immature female for queen triggerfish. All these purple dots are just primary growth oocyte, so really immature for sure for the picture. Maybe it don't make sense for me. I can help if somebody has the question. I can just help them later. And now I'm showing you a developing female. We have a different size of oocyte. We still have primary growth, but we have a bigger oocyte that is, and then we have a spawning capable female. That is what it looks like. The histology will help us to construct the seasonality plots. So again, this is Puerto Rico and St. Croix already published. Now we have a different looking for the red. So we see the red start in December. We read it that way. The red start in December, And then continue January, February, we have sample problem with the sampling, March through August. So also in St. Croix the same, the season start on December and then go through August. For St. Thomas, they are not yet reported. We also find the same, just only one change. We use spawning capable and actively spawning. That is a subdivision of spawning capable, but still the same. We have actively spawning females starting in December and continue through August. And the gonadosomic index, that is the way that we just measure the gonad and then divide it by the weight and multiply by 100, give it an index. The peak of St. Croix was February, the peak on Puerto Rico was December and January, and the peak for gonadosomic index in female was also January. And the L50 of female, the size of maturity, the L50 result from St. Thomas female was 240 millimeters, the fur length. Again, 50% chance at 240 that the triggerfish that you catch, if reef female, can be 50% mature or 50% or immature. So for resuming all this stuff, the maturity and the end of the presentation, we just have again in yellow the UNO, not the UNO, the St. Thomas data. That is the one that never has published yet. And then Puerto Rico and St. Croix, we have the L50. The L50 with their confidence intervals. And again, we have also in the immature column, we just include the N equals to a number is just the number of immature fish that we have. In St. Thomas, the males only have one, but we have to double check if that is an immature male or not. It is not. I just remove that value because I run the equation and the analysis doesn't give me a value because I don't have any immature, so they cannot compare. They cannot make a proportion. With this graph, I just want to show that we need just more data. I mean, it's a really data limited species, but we still need more small class size fish, and also St. Croix also have like a place size preference fish. So we also need the bigger ones and the smaller ones to try to complete the picture for all the 3 islands. And I don't have anything more to say, just thanks to the people, Rick Nemeth, Dr. Honey, Sarah Thomas in UBI, Dr. Sherbet and his lab, all the people in Fisheries Research Lab in DNR, the Nature Conservancy in St. Croix, Dr. Brendan Turley at University of Miami, and Nitin Ravikantachari from USC for helping me with the R codes, and the Puerto Rico and St. Croix fishermen, and also in the presentation is Julian, that is what really helpful. Without him, we cannot finish this sampling. And Marmat and the people on the Marmat and our funding. So any questions?
Thank you, Jesus. Jesus, we're going to save the questions to do it through the chat. Please pay attention on the chat to answer the questions because we're really tight on schedule.
Okay, cool.
Okay, and thank you very much. The presentation was great. Graciela, you have somebody to present? Can you help me?
I think we have Jennifer Moore online to talk about the critical habitat designation for corals and the big coral reforming basis of the fisheries here. So Jen, are you online?
I am. Can you hear me?
Okay, yes, perfect.
Okay.
So who's driving the presentation? You are, or?
I think I'm going to just share my screen.
Okay.
Are you seeing my screen? My presentation?
Yes.
Okay.
Make it into a slide mode and you'll be ready to go.
Yep. Okay. Great. Well, thank you for squeezing me in. This was kind of a surprise for us that that the rule published when it did. So sorry we didn't give you much more of a heads up that this was coming. My name is Jennifer Moore. I work with NOAA Fisheries in the Southeast Regional Office in the Protected Resources Division, and I've been working on this proposed critical habitat for 5 Caribbean corals for about 6 years. If you remember, I'm sorry, that should say 2014, that we listed 5 Caribbean corals as threatened. And we proposed this critical habitat on November 27th of this year. We have a 60-day public comment period that is open, and it will close on January 26th. And then based on our statute, we should have a final critical habitat 1 year from the date we proposed it. So that would be November 27th, 2021. Just to take you through kind of how we designate critical habitat. Critical habitat is required by the Endangered Species Act for all listed species unless it wouldn't actually aid in their recovery. So we go through this stepwise approach. And the first step we do is we identify the geographical areas that are occupied by the species at the time that they were listed. Then within that area, we identify physical and biological features that are essential to their conservation. What are the things that the species need to support all of their life history so that we can recover the species, and we protect those features within the habitat. We also have to determine whether those features actually require special management. There might be things that are part of the habitat, but that there are no activities that would adversely affect them, so they don't actually require any special management. And then based on those things, we identify the specific areas that contain those physical and biological features, and we map them. So to start off, we look at the geographic area occupied by the species. These species are present throughout the wider Caribbean. So basically anywhere there's a coral reef in the Caribbean is where the species occur, and that is the geographic area occupied. It's not every single location under which the coral actually resides, but basically geographic area occupied means the range of the species. However, critical habitat is a US regulation and only can be designated in the United States. So while the species are present throughout the wider Caribbean in the 30 so nations of the Caribbean, we can only designate critical habitat within the jurisdiction of the United States. So the real meat of critical habitat is the physical and biological features. And in this case, we have identified that the main recovery goal for these species is facilitating reproduction and then supporting their survival and growth after reproduction. So we've identified the physical and biological feature as reproductive, recruitment, growth, and maturation habitat. And there's a lot of words here on this screen, but basically what it boils down to is the hard substrate that the corals need to attach to grow and live out their lives, and then the associated water column over top of those areas of hard substrate. And then we further describe those features by identifying these kind of attributes that increase the conservation value. Basically, what are the qualities of that habitat, that hard substrate in the water column, that make it good habitat for corals to grow and survive? So in terms of the substrate, we're looking for things that would promote a successful recruitment of the coral larvae, so crevices, presence of crustose coralline algae. Also, those reefscapes have to be where the coral is actually going to be attaching and growing, need to be relatively free of sediment and macroalgae. Also, the waters have to have certain characteristics of ranges of temperature, aragonite saturation, which is related to how much calcium carbonate is in the water, levels of nutrients. Corals thrive in relatively low nutrient waters, and also have to have relatively clear water. So those are the things in the water column that basically support corals. And then in the absence of contaminants, we know that there are lots of contaminants that affect corals' ability to grow and reproduce. And so waters that lack those things are what are going to actually support recovery. So this is the really the meat of critical habitat is defining this biological and the physical and biological features of the habitat. And this is what we look at when we look at what the impact of critical habitat is what might adversely affect these things. We defined where that this is the feature that we want to protect through critical habitat designation. And then we go on to identify the specific areas that might contain those essential features. What you may be familiar with the Acropora critical habitat designation, that critical habitat designation is still valid, and nothing changes with that designation as with the new proposed critical habitat. But in that designation, we have one critical habitat unit that is for both of the species, for both elkhorn and staghorn coral. Whereas in this new proposed critical habitat rule, we are actually identifying 28 individual units, basically one for each of the 5 species in the locations that they occur. So for example, pillar coral only occurs from about 1 to 25 meters depth. So the boundaries are really one of the set of the boundaries of critical habitat are those depth contours. And then we look at the US geographic distribution. They don't occur north of Lake Worth Inlet in Palm Beach County, Florida. So that's the northernmost boundary. And they do go all the way out to the Dry Tortugas. And they are common within all of the waters of Puerto Rico, the USVI, and Navassa. So in those depths from 1 to 25 meters is where critical habitat is designated for pillar coral. And you can see that each of the individual other corals have their own depth distributions and particular geographic distributions, mostly that vary in Florida. The geographic distribution doesn't really vary in the Caribbean. Really in Florida, it's just about how far north they occur along the Southeast Florida coast. So I'm just going to run through the maps real quick. This shows you the maximum extent of the new critical habitat designation. Basically, this is for our corals that occur from half a meter out to 90 meters depth. And that's a couple of the Orbicella species and Mycetophilia ferox. So this is the maximum extent for Florida. This is the maximum extent for Puerto Rico, St. Thomas and St. John. You can see here that this is a single unit for these 2 islands because of those depth contours. However, if you were to look at the map for Dendrogyra for pillar coral, for example, you would see that there would be a break in the units between St. Thomas and St. John because of the deeper depths between those islands. And oops, sorry, I skipped over. Here's the maximum extent for St. Croix. And then apologize for the poor GIS data, but this is the maximum extent for Navassa Island and the Flower Garden Banks, which are off of the coast of Texas. But this is something you probably are more interested in is, what is the difference between the existing Acropora critical habitat, which is from 0 to 30 meters, and the maximum extent of this proposed critical habitat, which is out to 90 meters depth. So you can see that there are some areas that are new as compared to Acropora critical habitat. But what is not new is the fact that basically we are designating the ranges of these species. So basically anywhere that the species occur, we have been having to consider them in our federal Section 7 consultations since the time of listing. So really there isn't that much new that we have to really think about in a new consultation, because we would have to consider the species that were listed in these areas outside of Acropora critical habitat all along. So that takes us to kind of what I've been alluding to is what is the effect of critical habitat? Well, when you have a listing, there are individual prohibitions against doing things to the actual species. So since the corals have been listed in 2014, there are things that can and cannot be done with regard to the corals that might affect private citizens. However, critical habitat's only effect is via that ESA Section 7 interagency consultation with other federal agencies for their activities that may affect the proposed critical habitat. So in looking at those physical and biological features that we identified, we then consider, well, what are the federal activities that are either funded, authorized, or carried out by a federal agency that might affect the critical habitat? And so here's a list of the activities that we have identified that would, may be authorized, funded, or carried out by a federal agency that may affect the critical habitat. However, when we look at the impact of critical habitat, we have to consider what might be an incremental impact above and beyond the baseline. So again, as I alluded to before, the ESA requires Section 7 consultation on federal activities that may affect the species. Well, because the species occur throughout their range, we have been having to consult on the impacts to the species since they were listed. And basically, because corals themselves are their own habitat in many regards, the same activities that affect the corals would also potentially affect their proposed critical habitat. So many of these things we would not require a federal agency to change their activities based on the new proposed critical habitat designation because of the fact that the species occur in those same geographic areas. Also, we do have a substantial overlap of the new proposed critical habitat with the acropora critical habitat, and there is a general agreement between the essential features. So again, those things that we might ask federal agencies to do differently because of the proposed critical habitat, we would have already asked them to do differently because of the listing of the corals and because of the acropora critical habitat. So when in kind of going through that thought process, we identified that protected area management, fisheries management, and aquaculture, there would be no new consultations that would be triggered solely on the basis of this proposed critical habitat. It's not to say that you wouldn't have to consult on this proposed critical habitat, but these particular categories of activities all would have to have considered the species, acropora critical habitat, and those project modifications that we would ask federal agencies to do would stem from likely the listing and the existing critical habitat and not solely from the proposed critical habitat. So that's really how critical habitat rules impact the public, is via those Section 7 consultations, and how we might ask a federal agency to change their activities that they either fund, authorize, or carry out because of the critical habitat designation. I'm sorry, I slipped over a slide here. I wanted to also explain to you that there are some parts within those big maps that are not critical habitat. So I know those boundaries can scare people because they're really, really large geographic areas, but we don't have the data to map exactly those essential features that I described very, very precisely to inform the public exactly where they are. So unfortunately, we have to draw the maps with these large areas, but within those areas, the only thing that is critical habitat is where those essential features exist. So for example, if you have a large seagrass bed, that is not critical habitat because you don't have hard substrate. So basically what is not critical habitat is where the essential feature is not present. So that's the one thing that you do when you kind of look at the map and you kind of subtract out what is not critical habitat. Also, because we have a provision in the ESA that says we cannot designate critical habitat when there is a integrated natural resource management plan at military installations that would provide for the conservation of the species. If the things that they're doing to take care of their facility have benefits to this listed species, we are not able to designate critical habitat there. So we have one military installation at the Naval Air Station Key West that is not designated as critical habitat. Additionally, we basically, we want to only designate critical habitat for those things that are going to support the conservation of the corals. So managed areas, things that are like dredge navigation channels, shipping basins, vessel berths, anchorages, things that are constantly disturbed and would not provide good habitat are not part of the designation. Similarly, artificial substrates like aids to navigation, seawalls, boat ramps, you get the idea. These things do not provide the features that we need for the coral. So those things are not part of the designation. And lastly, we have one exclusion on the basis of national security impacts, and that's the South Florida Ocean Measuring Facility, which is off of near Fort Lauderdale, and it's a small area that is carved out of critical habitat that is not designated as critical habitat. So if you have an activity that either falls solely within one of these areas or is only affecting one of these types of substrates, like if you were, say, putting in a new mooring ball, or you were maintaining a mooring ball, then that activity would not have any impacts to critical habitat because it's actually not critical habitat. And so we wouldn't have to have you change your activity at all because there wouldn't be effects to the essential feature. So that that basically takes me through the things that are and are not critical habitat and how it may impact the public. Obviously, we are accepting public comment through January 26th. The docket number is there. So if you go to regulations.gov, you can submit your comment using that docket number. And if anybody has any questions, my email address is there on the screen.
Thank you very much for the presentation, Graciela.
No, that was Jennifer.
Yeah, I thought you want to comment something?
Marcos,
No?
because of the time, the question can be addressed to her at the email that she has there or in the chat.
Yes, we're gonna move along, and thank you very much. Pay attention on the chat, Jennifer, for any question that people might have.
Okay,
thank you very much.
Then we need 5-minute break, and then we go to Alida's report.
5-minute break, and we come back at 10:41.
Okay. Natalia.
Alida, Natalia no está, Elias.
Ajá, ok, Liajay. Tú tienes lo que Cristina mandó casi ahorita.
Sí, tengo una versión modificada
Ok.
que la envió por la mañana.
Pero tiene el
Tengo,
mismo número de slides.
te digo ahora, tiene 6.
Sí, está bien, está bien.
Ok. Y tengo una parte de Nicole Griot.
Sí, y Nicole debe tener también poquitas porque ella lo único que hizo fue cambiarle la primera,
Sí,
no cambiarla, sino ponerle que era el liazón de St. Thomas.
Claro que sí. Nicola están aquí.
Hola Nicole,
Hola,
me
Nicole.
alegro
Hola.
oírte. ¿Cómo estás?
Están, bueno, están muy interesantes de las presentaciones.
¿Verdad que sí? Qué bien. Pues nada, Leajay, yo voy a tratar de ir lo más rapidito posible. Voy toda mi presentación, entra la de Cristina y después yo termino ahí y entonces entran los liaisons. Me parece que así es que está organizada, ¿verdad?
Así es.
Entre todos tienen media hora.
¿Media hora? Ah, pues eso me da, muchacho. Sí, está bien.
Ok.
¿Qué hice? Dios.
Alida, si me escuchas, ¿puedo hacer una prueba de sonido? Te habla Guillermo, el intérprete. Yo.
Sí, te escucho.
Excelente, muchas gracias.
Ajá.
Y tomo la oportunidad para enviarte un caluroso saludo, amiga.
Igual, un abrazo grandote, grandote.
Qué lindo.
Saludos, habla Marcos, ¿todo bien?
Muy bien.
Todo bien. Saludos, hermano.
Un abrazo, gracias por tu ayuda.
Igualmente a ambos.
Alida, vamos a empezar. We're going to start with the presentation of Alida Ortiz, Outreach and Education Advisory Panel. Alida, go ahead.
Okay. Good morning, everyone. And before anything, I want to wish you happy holidays, happy new year, and the next year may be a little bit different from what we have right now. I will try to go through my presentation on the Outreach and Education Advisory Panel as so fast as I can. We have a lot of information to give you. Next, please. Okay, there were a lot of meetings that we attended as part of the Outreach and Education Advisory Panel, and it was from September through November, and we had meetings for webinars. We had another Outreach and Education meeting, and then we have meetings with the DAP, with the liaisons. Then we have meetings with UPRC grant to develop materials, and we also discussed the stakeholders' engagement with the LEMFES group and the Pew Charitable Trust. Then we had also meetings with Wilson and Christina, and we work with the webinar for tourism, the Puerto Rico Tourism Company. Next, please. I would like to just make an update on where we are with the Sustainable Seafood Consumption Campaign, which I think it's probably the widest and even the more aggressive campaign that we have ever had. First of all, the calendar of 2021 is also in the direction of how to promote this underutilized species. We did that with the 2020 and then the 2021. And we're also working on the recipe book that I gave some information in the previous meeting. We are already working with the recipes, the introduction, the catalog of the fishes that are used in the recipes. And I think this is going to be probably one of the most important products that we will have for sustainable seafood. Next. The webinar that we gave for the tourism company to me was very important because it is bringing the sustainable seafood campaign to the wider people because the tourist guide or the tourism guides are the people that take the tourists to the restaurants, to the places where they sell fish. So they have to know about this campaign. And they were very interested. Marcos Hanke and myself made the presentation. And from that presentation, a group has been formed, a working group with the tourism company where we're going to develop other activities and see different ways of reaching all the audiences that we can. Next. We're also working on short videos that Janet Ramos and Cristina Olan are producing. Probably Cristina will speak a little bit on them in her presentation of the social media. But these are videos directed to the consumer, to the person that cooks in the house, and they're very simple, very easy to follow, and very attractive. And then we would like also to develop a guide to analyze those underutilized species. How can we use them in educational products? How we can have people learn about those species that are available, that are edible, that are easy to cook, and would take a little bit of pressure from those species that we use historically all the time. Next. We're also working with materials for the marine fisheries ecosystem of Puerto Rico, and the Virgin Islands, so we can make a wider distribution of that. And one of them is a new short video, one-shot video of the habitat of particular concern, utilizing the information that we have already on the essential fish habitats. And this is just to bring to the public the importance of knowing what they are doing in the coast or in the mountain that will affect those essential fish habitats. So these videos are being produced by Ambiente, and they will be available in the social media as Christina received them. And then I'm trying to work on a request that I had from Nelson Crespo, which I'm very, very grateful. How can we produce the materials from the book for the 4-H groups? So I'm working on the slide presentation on the basic concept that we discuss in that book, so that the 4-H that are still meeting virtually can use them. Next. The other project that we're working on, outreach and education, is a series of posters. The first one is a review of the poster on the life cycle of the queen conch, and you have it right here, what it consists of. The text has been reviewed, updated, and also the art. This is just about to be finished, and we are very grateful for the support that we have received from Dr. Richard Apperdon, from Graciela Garcia Moliner, from Miguel Rolón, and from the people that have seen it, Dalila Aldana, that have seen the text, and they have sent us their recommendations. This one we will send it to Miguel for printing early in this December. Then other posters, give me the next one, please. The other posters that we are working also are posters probably in the similar approach that we have for Naso grouper and from mutton snappers, with Spanish, English, and French versions, because they were going to go to the international campaigns where the U.S. Caribbean Council the Caribbean Council is working. It will have the biological aspects like life cycle, prey, predators, spawning, aggregation, seasons, and all this in a very good illustration, but also with very easy to understand language that they can, that the fishers and the consumers can get the information. Next. We are working also on outreach materials on protected marine areas in the USBI. This is in response to a meeting, a very, very important meeting that we had with the DAPs, with the local government, and with the liaisons in the Virgin Islands, so that the marine protected areas like the Germanic Bank, the MCD, and probably with the parks and monuments, all those areas that are protected for the fishes, for the species that live there, we can have information in form of fact sheets, posters, stickers, wallet cards, short videos for social media, so that the tourists and all the people that go to this area recognize the importance of following the regulations, or respecting, the habitat that is there. That is very sensitive, but it is also very, very important for the entire fisheries in the Virgin Islands. At some time, we may do the same thing for the Puerto Rico areas in the Bajo de Sico and those areas where there are regulations for fishing. Next. So we would like to open it now for Christina to give us the information on the social media. Please.
Good
Alida,
morning.
Before, Cristina, before you go, excuse me, the last slide that was presented by Alida responds to a request by Fisher from St. Thomas and John Julian and Tony Blanchard and Ruth Gomez, who were at a meeting with us. So once we have these materials, we want them to check what we produce just to make sure that we cover the items that were of interest to them. And then we will go ahead and make it a final product. And this proposal is between Sea Grant and the CFMC with the collaboration of Dr. Ali Ortiz and others. And it will go through CARICUS, which is the entity that will be allowed to manage this proposal and the proposal for St. Croix. Thank you.
Actually, we have a meeting with them it's the next week, and we will discuss the proposal.
Okay. Sorry, Christina. Thank you.
Thank you. Good morning. My name is Cristina, and I work for the Caribbean Fishery Management Council as social media content developer. And thank you for the opportunity of presenting what we are doing for social media. Natalia, next slide. We continue publishing information on seasonal closures, announcements of webinars offered by other agencies and organizations, for example, NOAA bulletins, the announcements of our meetings, species. We have been producing slideshows about species that have seasonal closures. We also publish information related to essential fish habitats. We also share the administrative orders from the DNR and also communications from the DPNR, especially during the pandemic. We have been sharing Amanda Oceano Facebook Lives, especially the ones related to fish identification and lionfish. And also I want to thank to all the persons that are always sharing the information and providing their input for our posts, especially to the CFMC staff, fishers, scientists, agencies, and stakeholders. Next slide. A couple of months ago, in September and in October, we broadcasted 2 Facebook Lives where we explain the use of the voting app developed by CARICUS. It was an effort among CARICUS, Puerto Rico Sea Grant, and the Caribbean Fishery Management Council. Next. In YouTube, we have now the recordings of the DAP meetings and the regular meetings. We also have a video on ocean acidification that was funded by the NOAA's Ocean Acidification Program. And also we have the recording of the Responsible Seafood Consumption webinar sponsored by Puerto Rico Tourism Company, where Alida and Marcos were the presenters. Next. We have also been producing a monthly bulletin. We publish it on November and December. And the bulletin includes information regarding CFMC meetings, underutilized species, fishers, sustainability, information in our social media platforms. The bulletin is available through Facebook, the CFMC webpage, and email. If you have questions, please let me know. I'm thankful for all the recommendations and suggestions that our stakeholders, fishers, agencies, and other persons are always sharing with me to improve the content that we share through our social media platforms.
Thank you. Thank you very much, Christina. Next.
Yes, I'm sorry, Alida. I also want to mention that in October, that was the seafood month, we also published a couple of recipes that were provided by Janet Ramos. And it was a collaborative effort between Janet, also Michelle Shire, that sent us scientific information, Efra Figueroa that shared footage of the pejepuerco, the triggerfish. So we published just 2 recipes, and also we share information related to seafood consumption. Thank you.
Thank you, Christina. Great work. And Elia, how can we go back to, okay. So as a recommendations to the council that we have now, this outreach initiative to educate the public on the values and importance of the MPAs for a sustainable fishery is very important. We recommend that the council consider support for all the initiatives that we are taking now in the USPI and to promote an extension of those same type of materials for the Puerto Rico protected areas. They are needed. People have to know where are the areas protected, why are they protected? And with the responsible consumer and sustainable fishery outreach initiative, to us it is very important because it involves the consumer in the fisheries protection. The protection of that resource is not just the responsibility of the fisher or the managers, it's responsible of us as consumers. So we would like to extend this initiative from that we are doing in Puerto Rico to the USPI, and we request the support of the council for these projects. Next, as we decided in the meetings that we have with the liaisons in the last month, I think it is important that each one of the liaisons, these are the people that connect outreach and education with the fishers, with the community where they live. So we have Wilson Santiago from Puerto Rico. Next, we will have Nikita Charles from St. Croix. And after that, we will have Nicole Greaves from St. Thomas, St. John. Liahai, you can put them now. Wilson?
Hello, good morning to everyone. Wilson Santiago here, liaison officer for the Puerto Rico Island. Yes, you can pass the other one. Okay, the 2020 liaison participation. So I have been coordinating the PEPCO program, the virtual PEPCO program, resources and presentations. I think I'm gonna finish everything with the PEPCO program in December and planning to start in the end of January 2021. Okay. The other thing is support Cristina Olan with the new post of the CFMC social media regarding closures and DNR administrative orders for Puerto Rico fishers. And I have participated in different workshops regarding fisheries education.
Toca la tierra. Ah, ok.
Okay.
El micrófono está abierto, Álida. Adelante.
Okay, thanks. Okay, I start making a database of the participant of the program PEPCO, where there are telephone numbers, emails, and postal address, and so I haven't finished past them. Or right now I have around 430 contacts. So that database will work for the
Hold a second, Wilson. You know, please shut your mic because we are hearing people doing the dishes or something. So if you're not talking, close your mic. Thank you.
Okay, thanks, Miguel. So this database I was talking about, it will work with the outreach of the council, the outreach of the DNR, and any other agency that need that database. So when it's finished, I will send it to the CFMC and to the DNR so we can use it. Now this database, I started making it because of the in the last meeting, one of my proposals was making like a push notification message, so we can weekly or monthly, we can send notifications regarding educations to all the fishers and fishers community about closures, everything regarding the fisheries. Okay, so I'm planning to in 2021, so we can start this push notifications. Okay, also have been supporting the CFMC, finding pictures and area of catch per species for the 2021 CFMC calendar. I have been supporting Fisher with issues and information of the DNR state and federal closures. And having given educational materials for the fishers, educational materials from the CFMC. Okay, next one, please. As a liaison officer of Puerto Rico, one of my commandments are taking the issues that are regarding the fishers in Puerto Rico, and like right now in the pandemic, the majority issue within the fishers in Puerto Rico has been the license and permit procedures of the DNR. I have been working with the DNR Office of Permits, so I can be updated weekly. So when the fishers call me, so I can tell them in what stage are their permit or license. That is like the most issues right now in the pandemic for the fishers. Okay, I also have seen the new features. So right now the DNR have around 2,000 solicit the new fishing permit. They are new, so there's a lot of misinformation about the fishing closures, statistics reports, license and permits for state and federal waters in Puerto Rico. So I think we can outreach those features and educate them with the PEPCO program. So that is one of the tags that we have with the PEPCO program. Okay, and the other issue, the fishers in Puerto Rico shown that we have this issue like every years, is the law enforcement to watch the closures and illegal commercial fishing in state and federal waters. So that is all for me. And if you have any question, you can send them via the chat. Okay, thank you.
Thank you, Wilson. Alida.
Hi, good morning, guys. First, I'd like to thank everyone for having me. My name is Nikita Charles, and I'm the CFMC liaison for St. Croix. You can go to the next slide. As you know, right now we're working on the Reef Responsible Sustainable Seafood Initiative, and we've been dedicated to having the program up and running again at full capacity by the end of the year. And our goal for the initiative is to promote the Virgin Islanders to catch, purchase, serve, and consume locally harvested seafood. Reef Responsible wants to work with the community through engagement and education of commercial fishers and restaurants to make better choices in seafood consumption. Next. In terms of educational material, we've recently gotten our posters and brochures. We've worked with the Department of Fish and Wildlife to get the educational materials printed out, for like the fish fact book and whatnot. And with that, we're going to be dispersing and promoting and educating the community about the program. In terms of social media, as a team, we've established incorporating weekly posts on social media outlets such as Facebook and Instagram to help promote and bring awareness to our mission. And shout out to Danielle Lee from St. Thomas for helping us add items such as Trivia Tuesday and Fish Spotlight Friday to our pages, promoting the educational material such as the fish fact book. And then for commercial fishers, it's actually this coming Friday, we'll be welcoming fishers at the reopening of the Lorraine Fish Market and giving them the opportunity to learn more about how we can help them and how they can sign up for the initiative. And here we'll also establish a voluntary list for them to be able to have a list of licensed commercial fishers that we can share with the restaurants in the future. The next slide. In terms of restaurant trainings now, we've added more restaurants to the list of responsible restaurants for outreach. We're currently scheduling restaurant trainings for the month of December. We've also been working on establishing monthly Zoom trainings because, as you all know, there's COVID. And we're trying to add a presentation that works interactively virtually with individuals, you know, so that we can ask them questions and that they can participate better. And, you know, we have our Reef Responsible Advisory Council that we've been utilizing, and we've gotten input on the presentation. And, you know, just to give you an idea of what the rest of the training material for the restaurant trainings and soon fishing trainings, we decided to add things like the stony coral tissue loss disease and exhibiting ways to properly measure legal sizing for purchasing specific types of catch, that being adding how to measure and better visually explain the carpus length and fork length, how to get a better idea of telling if a fish is fresh, you know, if you're getting fresh fish, making sure you're purchasing from licensed commercial fishers, because I know that's a big deal for a lot of people, and how to check for that. And then there's discussing the high risk of ciguatera. I think that's all I have for you guys today. Thank you.
Nicole? Nicole, you are muted.
There we go. Good morning. Can you hear me now?
Now, of course.
Okay, fantastic. Good morning to everyone. Thank you so much for allowing me to do my presentation at the Fishery Management Council. My name is Nicole Grillo. I am the liaison for St. Thomas St. John. First off, I'd like to start with saying we've had some challenges over here on St. Thomas. I lost the expertise of Alexis Sabine upon her resignation. So we've really had to start off fresh over here as far as obtaining information and getting things like fishery contacts and those kind of items. Next slide, please. So we're starting off with the fishing vending areas. One of the issues that I've come across speaking to fishermen at our 7 vending sites, believe it or not, is that they really would like to know a timeline for repairs and improvements on a lot of the fish vending areas. Only 2 of the fish vending areas that we have here belong to Fish and Wildlife or DPNR. The others are more traditional than designated. And that is going to be a discussion I know that's going to be soon coming. I do believe that Julian McGrath at the DAP meeting had spoken about getting in contact with some of the owners of the properties that are now traditional vending sites. Next slide, please. So representation in events for the fisheries. I've noticed that we have not had a very strong representation for our local fishers here, the commercial fishers in the Virgin Islands. We are hoping through the Reef Responsible program to have more of our local commercial fishers out and about so that people can know the key part that they play in our fisheries and also in helping to get information to the public as far as the different species of fish are concerned, and also what species of fish are considered the most desirable. They're also the ones that have the most information on things like fish growth, fish availability, and also as far as the ciguatera areas are concerned. Next, please. So as far as the liaison and fisher discussions go, we have yet to have a full meeting. Obviously, COVID has put a damper on a lot of our meeting capabilities. And as far as virtual meetings go, some of the fishers are not very much into having virtual meetings. So that's going to be put on hold until we feel more comfortable meeting in public. I mentioned earlier that I went to 7 of the fish vending areas. I have been visiting the fishermen and their helpers. They are quite a very amazing group of people. They have so much resilience. I'm very proud of our local commercial fishers, both here in St. Thomas and in St. John. Another thing that has come to my attention while I was going out to meet the fishers is that there's not very much information that is out for them right now as far as their hurricane relief funding is concerned. And I am going to ask, since I know that Dr. Anjali is here on this call, if there can be something that you and I can discuss as far as putting together information that I can go ahead and hand out to the fishers, or that we can get together and speak to them about so they can have those questions answered. The Reef Responsible Initiative that Nikita was talking about is going to be our main platform for the Responsible Consumer Campaign. I'm very proud to be a part of that particular initiative, because not only is it going to help people learn more about what sustainable seafood is, it's also going to give a better scope of the fishers available to not only our restaurants, but also to our local consumers of different fish. And that's all I have for now. Thank you.
Next one, Leah. Hi.
Benito.
Thank you so much, especially to the liaisons. I think the work is magnificent, and I ask you to keep contact with us, not just for the meeting on the council has, but every time that you have an activity, send it to Christina, send it to me, so that we can, share how this connection with the fishers is working. So thank you so much. So this is all our presentation. If there are any questions related to outreach and education panel, yes.
Farida, falta la presentación de Cristina, que tiene que ver con outreach and education, de los trabajos que se van a hacer científicos alrededor de Puerto Rico y las Islas Vírgenes.
Okay, I didn't have
Cristina, are you ready with that?
Yes, I am. I have my presentation, 3 slides here. If I can share the screen.
I didn't know that.
Okay. Are you able to see it?
Yes.
Yep.
Okay. The title of this proposal is From Fishers' Knowledge to Scientific Language, Understanding Essential Fish Habitat of the Deep Water Snapper Fishery. And the objectives of these efforts are the following. Document what fishers do in this fishery and how they contribute to science. Create awareness about deep water snapper fishery and the essential fish habitat associated with it. Increase the understanding of this fishery and inspire stakeholders to support management measures to protect the fishery and promote sustainability. This proposal has 3 phases. The first one that we will be recording virtual interviews with scientists that are working with deep water snapper fishery, and we will be producing short videos for YouTube. The second one will be interviews to fishers that are collaborating with research to highlight the importance of their contribution and their knowledge to science. Those videos will be published in YouTube, and also we will be working on some bites for Facebook and Instagram. In this part, we are going hopefully to be interviewing fishers in person instead of doing virtual interviews. And then we are going to produce a video of deep water snapper fishing, or in Spanish, cala. That's my presentation.
The other thing that we have here is that the monies for the presentation, I mean, for this proposal come from the funds that were approved by the Habitat group and the Reef Fish group from NOAA, that Graciela monitors. And this money has been approved already, and it will cover, of course, the 3 islands. Remember that we have the island-based FMPs, and it will be implemented accordingly in 2021. However, this effort will include all features from St. Thomas, St. John, from St. Croix, and from Puerto Rico, of course. The scientists that we are going to interview include the 3 presenters today, and all the presenters really that have anything ongoing in terms of research or have done research in the Virgin Islands and Puerto Rico that will be of interest to the fisheries of this area. And the interviews in the field will depend on the COVID guidelines. And as they told us yesterday, National Fisheries Service is still on phase 0 until the end of the year. They have several phases, but we envision that probably by the end of 3rd quarter of 2021, we will be able to have the vaccine and allow people to go in the field. The proposal will cover probably 2021, 22, depending of this COVID thing, and also the timing of interviews with fisher and so forth. The last part of the proposal includes a video on how to fish deep water snappers and groupers. This idea came from conversation with fishermen in the U.S. Virgin Islands and Puerto Rico, and it will be a fisher-to-fisher talking. We have, for example, ex-fisherman from Puerto Rico explaining how he or she fishes for deep water snappers and grouper, the experience and all that. And then we will move to Virgin Islands and see who in the Virgin Islands fishes for deep water snappers and groupers. And that will be posted and available in Facebook for any fisher who would like to venture into deep water fishing. It's not an easy fish to do, an easy fishing to do. Otherwise, everybody will be doing it. But we believe that this is an answer to many fishers who requested more information on how they can go into deep water fishing. And it is a commitment that we made at the beginning of the year with fishers, and this way we will be able to fulfill that. Alida, do you have anything else that you want to add?
If I may, Miguel, before you.
Go ahead, Graciela.
So the Coral Reef Conservation Program provided the funds to the council to actually follow the fishers as they do their fishing, and to answer specific questions regarding temperature at depth, the water masses where they're fishing. So it's not only the component of how they fish and the knowledge that they have in selecting the areas and the depth for the size of fish that they are targeting, but also the oceanography of that data to bring their knowledge into the science realm. And I just saw in the text that Jesus, Kate, and Virginia would be very happy to be interviewed. We had sent some emails around to make sure that we encompass both the scientific efforts that are being conducted and the actual description and characterization of that fishery as they prosecute that fishery. So, the key feature is their knowledge being translated into scientific language, and to answer specific questions that they have regarding the changes that they notice in their fishery, for example, temperature. So we are using a CTD at the same time that they are fishing to get environmental data and to look at the parameters that might be impacting the size of the fish and the changes that they notice from they go to the same area over time. So, this is really exciting that everyone is working at the same time to include life history information and actual data from the fishers to really characterize this deep water fishery. So thank you to everyone who's participating.
Thank you, Graciela.
Okay, Miguel, I don't really have nothing else to add. Just say thank you to all the collaboration of the Outreach and Education Advisory Panel, that they keep us in touch, and we will work closer with the liaisons. And it is important, this production that is being done for the social media, because we have to reach the people from every possible way that it's available. Thank you so much.
Thank you very much, Alida. Miguel, do you need anything from the council or
No.
just the presentation?
No, in the case of the presentation by Cristina, well, we wanted to, you know, if everybody agrees, we will continue with the presentation. We had the funding, the proposal that was approved, monitored by Graciela, called for the social media person to be involved, and that's Cristina. Given that the money are beyond what we have in the purchase order, we just want to make sure that the council understand that this is what is going to be done. And if there is no opposition, we can continue with the project, or you can have a motion to continue with these efforts, and also to keep collaborating as suggested by Dr. Ali Ortiz to move forward with the project that we have in Puerto Rico into the U.S. Virgin Islands. And that's more or less what we wanted to. So either way you do it, it'll be fine. You know, if you want to have a proposal or a motion to make it stronger, that would be appreciated.
Yes. Do you want, can you help me with the language of the motion?
Okay, the motion will be to accept the report from Dr. Ali Ortiz and Cristina Olan, and to support the proposals suggested for outreach and education with the participation of the U.S. Virgin Island Fishers scientists, as well as those in Puerto Rico.
So any of the council members would like to present that motion?
Just say so, I move, and second.
This is Nicole Angeli, so I move.
Have to be a council member?
Nicole Angeli is a council member.
It's a council member.
Yes, I couldn't hear who spoke. I'm sorry, Nicole. Yes, a second then?
Second.
Any opposition?
Second.
Hearing no opposition, motion carries. Thank you very much, Miguel. We move on.
Yeah, thank you very much, Mr. Chairman.
The next presentation, it will be the enforcement.
Bless you.
You are very emotional with enforcement. The first one is from Puerto Rico. Damaris Delgado, are you going to do the presentation?
Yes, good morning. So for Puerto Rico, as you know, we have been, rangers have been aiding with the affairs related to COVID. But besides that, they still have been intervening with the regulation, environmental regulations associated to fisheries. And we have 35 cases of nets, cast nets, trammel nets, and crab traps in the Humacao region. And this information that I'm sharing with you is from January to November. I couldn't get the information from our last time, the last time that we report, but this is a global data on the interventions from the rangers from January to November. So we had 23 cases of cast nets in the region, in the Humacao region, 5 cases of trammel nets, and 7 cases for crab traps in that region. Besides that, there were several cases on beach seine. I don't know how to pronounce that. I say it chinchorro is the way I know it. So in regards to the chinchorro, we had 4 cases of that at the South region, a lot of interventions in the South region, including one with the beach seine in the river mouth of Nigua River in Salinas. There were 19 cases of interventions of hooks and lines in that same region, South region and the Southeast. We had 1 case of handline in Salinas, 5 cases of lobster pots in the South region. Several cases were of lobsters because of not fulfilling the size, the required size. We had 3 cases of interventions for not having licenses or permits, including expired licenses, but mainly because they didn't have license or permit. There was also 2 interventions for people violating the ban on crabs, 1 intervention for the closure and wahoo species in Cabo Rojo, 2 cases associated to divers. And that's pretty much the summary of the interventions. If you require the details, I have the report that was provided by the rangers with the exact cases and the number of the cases and the places where they were made. So that's pretty much the information that I have. Some of the interventions that the rangers are doing are being posted in our mass media, including one recent case of illegal fishing in Humacao. So that's pretty much the information.
Thank you,
Yes.
Damaris. Can you send us an email with an attachment so we can put
Yes.
it in our records?
Yes, sure. I'll do that. Send it to
And
you.
one clarification, most of these interventions are because they're in violation of Puerto Rico fishery laws.
Yes.
And regulations.
Right.
Thank you. Thank you
So
very much.
that's law 278 and regulation 7949. Thank you very much.
Thank you, Damaris. We go now with the Coast Guard.
Good morning, everybody. This is Lieutenant James Bruce with the U.S. Coast Guard. Can you hear my audio okay?
Yes, we do.
Brilliant. Thank you very much. Hey, I like I said, I'm Lieutenant James Bruce with the United States Coast Guard. Thank you very much for the opportunity to speak. I will be brief. It's been a great 2 days. Some of the presentations from everyone have been really, really interesting. I don't have a presentation. Especially after how professional the other ones are. I wouldn't dream of bringing one to compete. But the only thing I would submit, the U.S. Coast Guard, we're continuing to work with our partners and agencies from Puerto Rico and the area, USVI, state and federal partners. We're continuing to conduct enforcement patrols to enforce federal fisheries and federal regulations. COVID has definitely been an interesting year and presented some operational challenges for us to make sure that we are able to protect the people that we interact with as best as reasonable and possible, and also protect our crews while still conducting this mission. Other than that, I don't have a lot else to report. The U.S. Coast Guard, I mean, we stand ready to support the communities that we're involved in, and also try and help level the playing field in the commercial fisheries. So I'll stand by if there's any questions, but that concludes my remarks. Thank you.
Thank you very much. I skip accidentally the USVI report.
Thank you, Mr. Chairman. This is Nicole Angeli reporting for enforcement for the Department of Planning and Natural Resources. Our officers are currently pulled to the Virgin Islands Police Department in order to enforce and ensure the health and safety of our citizens during the COVID-19 pandemic, including enforcement for boating restrictions. The fisheries enforcement has no updates. However, we do have good news in that we have managed to hire 2 new candidates for the police academy this year. That concludes our report. If you have more specific questions, please let me know in the chat or email later.
Thank you very much for your report. The next report from enforcement is the NOAA Fishery Office of Law Enforcement.
Marcos.
Sí, adelante.
This is Miguel Borges, NOAA Fisheries Law Enforcement. I think Manny Antonaras was going to speak, but he might be dealing with another thing. So I could give a couple of updates, at least since the last council meeting in September. We've had several interdictions and trainings since then. And specifically in training-wise, we provided training for the DNER officers of the West Coast of Puerto Rico, specifically Aguadilla and Boquerón. The training was regarding case package requirements and federal regulations implementation of the closed areas in the West Coast of Puerto Rico. We also had a meeting with the Secretary of DNER and the Commissioner of DNER to implement the strategies for future collaborations coming up. In enforcement actions, we've had several interdictions that have been in collaboration with DNER officers that have resulted in enforcement action, and those have been regarding highly migratory species, specifically billfish. We also had an enforcement action concerning dolphin harassment in the east coast of Puerto Rico that also resulted in enforcement action for us. And we are also continue working with the Coast Guard and DNER for future operations. We are also working for the long-term investigations that are still ongoing. And lastly, we are focusing a lot of our enforcement efforts towards port state measures and IUU fishing. So that's unreported illegal fishing, and that's done through the seafood importation through the ports, mainly the San Juan port, and same thing for the USVI, San Thomas, for all the regulations to import seafood from other countries and the programs they have to abide by. So that's first time in Puerto Rico being done. And that concludes our report for this time. Thank you.
Thank you, Miguel. Thank you for your report. We are going to other business now. I believe we have a presentation from Carlos Farchetti, designating a safe fishing zone.
Yeah, thank you, Mr. Chair. I think Natalia has a slide for me.
Yeah, ahí tienen lo de safe zones. Thank you.
Okay. Thanks. Alrighty. For years, fishermen have been complaining about cargo vessels taking shortcuts over the shallow waters of Lang Bank on their way to the Port Authority container port on the south shore of St. Croix. These container and cargo vessels, while on the way, have traversed through Lang Bank at water depths of 40 to 60 feet, and while doing so, have entangled fish trap lines, dragging and destroying many fish traps, causing economic hardship to the fishers who have had to replace quite a bit of lost traps throughout the years. And who knows what damage to the habitat these traps have caused when being dragged by these ships down the road. In the 1980s and 90s, fishermen have taken shipping companies to court. A couple have been compensated by the company when fishers have identified their buoy colors. However, most of the fishers do not have the financial backing to hire attorneys to fight their case compared to the legal defense a multi-million-dollar company can afford. So really all the fishers can do is complain to the DPNR. This problem occurs predominantly on the south shore of St. Croix when ships traveling from the southwest cape of Sandy Point to the container port. In the past, there has been a notice to mariners on the coast pilot for cargo and container vessels to stay outside of the 100-fathom curve while approaching the container port. This notice was removed in 1995. However, I'm not sure why, and I couldn't find the reason why it was removed. I am aware that the area of the South Shore is in territorial waters because the 100-fathom curve falls within state waters. But I believe it's an important issue that not only occurs in Lang Bank. However, being the council and where we only regulate federal waters, that's why I'm specifically speaking to the area you see on the screen. When it comes to the South Shore of St. Croix, the fishermen met in October with the governor of the Virgin Islands, and he said that he would be consulting with the Port Authority to discuss what can be done about designating a safe fishing zone on the South Shore of St. Croix. These incidents also create a safety at sea issue, particularly at night when fishers are line fishing and while at anchor have had to cut their anchor line to escape being run over by these large vessels. This safety at sea issue also occurs at Lang Bank, and I believe some type of protection should be afforded to the fishermen who use this area to make their livelihood. And speaking to one of our DAP members, which is also a member of our FAC, David Gubser, he brought up what happened in San Francisco Bay while he was a tugboat operator. And they have had designated shipping lanes to avoid this problem. All that being said, I would like to get some advice from the council, maybe from Jocelyn or maybe the U.S. Coast Guard, on how we can designate the area of Lang Bank seen on the slide as a safe fishing zone by maintaining vessels outside of the 100-fathom curve while navigating around Lang Bank, St. Croix. Some of these vessels are either out of Puerto Rico or the U.S., somewhere in the U.S. or even down island. I believe a rule of law could be what the fishers are asking for, because placing a notice to mariners in the coast pilot as a courtesy, as it once was, is not good enough, and it's usually ignored. You know, if there's a way to have language such as vessel X measuring or weighing X metric tons must or shall keep within X, Y, Z rum lines is necessary language. If this request for assistance by the council is beyond its jurisdiction, I would like someone to maybe point me in a direction that I can follow through with it. Thank you, Mr. Chair.
Thank you, Carlos. You request the input, if it's possible, from the Coast Guard and from Jocelyn. Let's start with the Coast Guard. If there is any input, any light that you can help here, Carlos.
Hi, everyone. It's Lieutenant James Bruce with the U.S. Coast Guard. This in particular is not my area of expertise. What I did is put my email address in the comments. If you would be so kind to just give a quick recap, if you want to send me the the issue at hand, I can absolutely get you to the right office that would be able to speak to this with more authority and precision. So I hesitate to comment too much on it because, again, it's not my particular area of expertise. However, the process of these types of designations is something that the Coast Guard does participate in, along with several other federal agencies. So I'll pause right there and just make sure that that got through, because I know we're on a virtual meeting.
Yes, thank you. Matter of fact, tonight we have a Fisheries Advisory Committee meeting, and I will bring up this point that you just brought up, and we'll send you an email. We have a member named Tobias or William Tobias that has a long history with what's been going on out there, so he can assist me in writing this request to you.
And sir, it's James Bruce. It's Lieutenant James Bruce again. It doesn't have to be anything very official. What I'm more speaking to is just to get you on the right path and get you connected with the right people. So I just want to make sure I'm communicating that clearly.
Sounds good.
Carlos, probably we should do two things. Follow the Coast Guard suggestion is just to get the names of the person that you should address this request, and then put together a request, because I worked with this before in San Juan for another reason. And then the request, usually what they want is the why, where, the rationale, etc. And then they point to the other agencies, because this has to be done by the action agency, and you have to include a lot of considerations for that. So I guess that this is the best way to start. And then National Marine Fisheries Service intervenes with comments regarding whether any of this action have an impact on the fishery management plan and so forth. So we should send a copy to Jocelyn just to make sure that they are abreast of this development.
Okay, will do.
Thank you, Carlos. I think it satisfies what your intention is so far. You're done?
Yes, it does.
Thank you.
To start.
Yeah. Yes, thank you. Thank you. The next, I think we have another business. That's it, correct, Miguel?
Yes, we had the public comment period, and I believe that Dr. Michelle Shatter wanted to address the group.
Yes. Going now to public comment period, Michelle.
Hi. Marcos. Excuse me, Marcos, I'd like to make a comment and a public comment when she's finished.
That's correct. Thank you. No problem.
Bye-bye.
Hi, everyone. Thanks again for the opportunity. I believe there's a short presentation that I sent to Liahai and Graciela.
Yes, you did. Hold a second because your PowerPoint just froze. So give me a second to reopen it.
Basically, I just wanted to reiterate what various scientists shared this morning, that we need data from our local area to be able to adjust our management of our fisheries, since a lot of the information wasn't available previously. For example, these age validations and differences in the behavior of species due to our environmental patterns in the Caribbean being different from the US. So basically, I wanted to bring to light something that I've shared previously with different agencies and persons regarding the variability in the formation of the spawning aggregations we have locally. So this is preliminary data from our passive acoustic monitoring of red hind off the west coast of Puerto Rico. So
Michelle, can you tell me if what you were saying is your presentation?
Yes, ma'am, it is correct.
Okay, so let me know and I'll...
Next. So red hind off of western Puerto Rico are known to aggregate to spawn during 1 to 3 lunar cycles after the full moon of the winter solstice. So right now, today, December 9th, the fish are starting to migrate to their aggregation. But this year, the full moon is 29 of December. So it's not until after that that we'll actually see the aggregations. We also know from work done here and in the US Virgin Islands that the males and the larger females, which will be next year's males, because remember, they change sex as they get bigger, they remain until the end of each aggregation every year. And this is important to understand how the behavior is affecting the aggregations. We also know that one of the cues for spawning is the water temperature needs to drop below 26.5 degrees centigrade for them to actually have that cue to spawn. During the reproductive behaviors, red hind produce sounds that we can detect remotely by passive acoustic monitoring. And we've been doing that since 2007 at Abrir la Sierra. If the aggregation extends past February 28 in the EEZ of Western Puerto Rico, the reproduction is disrupted. And because of their behavior, the larger males and females will be more vulnerable to fishing after the close season ends. This happened 3 times in the past 10 years that we've been monitoring. If you go to the next slide, I can show you an example of what this data actually looked like. So the orange bars are the acoustic signals that are produced by the red hind when they are aggregated. On the top, you'll see the full moons of that season. And you'll also see dotted blue is the minimum water temperature at depth at this aggregation site. So basically that first peak in sound production coincides with that first dip in water temperature below 26.5 degrees. And then we see another peak in sound production that also comes after another dip in temperature. And that red dotted line is February 28. So we've been able to document 3 times in the past 10 years that the aggregation continues past February 28. Next slide, please. So we can see that the aggregation is well protected during the first 2 cycles, but it's not always protected when the full moon is late and the temperatures don't peak down into the colder areas that they need to spawn. Next slide, please. So why is this important? Preliminarily, we've been collecting red hind from commercial fishers in different parts of the island. And this is a project in conjunction with Rick Nemeth and Virginia Shervette, funded by Marfin. When we look at the sex ratios or the proportion of males to females off of Western Puerto Rico throughout the fishing areas, we see about an 80-to-20 ratio. On the east of Puerto Rico, it's a little more males to females. But during the aggregation site that we've been monitoring off Western Puerto Rico, we have a scarcity of males. And I don't need to explain much how you need sperm to fertilize all these eggs, but this is something that is concerning for the productivity of the red hind fishery off of Western Puerto Rico. Next, please. So the variability in the lunar cycle, so this is when the fish actually aggregate compared to the regulations that end on February 28. Based on preliminary data, we could see this happen up to 8 times in the next 10 years. But it's not sure how climate change and seawater temperatures will affect these patterns. The suggestion is to go to the CFR subpart S, section 622.435, and revise the end date of the red hind closed season if the rationale is to protect the aggregations. The future aggregations would be better protected by a more in-depth analysis of our passive acoustic monitoring data, a continued active passive acoustic monitoring, and year-round seawater temperature recordings at different aggregation sites so that we can address how widespread this problem may be. The variability in the formation of the spawning aggregations has also been noted for other species that aggregate to spawn in the US Caribbean, and we have that data available for the groupers that produce sound. Finally, next year in February 28, we expect the fish to still be aggregated after the closure of the red hind season. And we suggest that the council consider an emergency rule so that this aggregation is not disrupted in the near future. And that's it. Thank you very much.
Any question from the council? Even I think it's very important information that's relevant to all the regions of the US Caribbean. Any comments? Any question?
Marcos, I believe that what Dr. Shatter presented today is new information, or let's say presented in a different context. I believe that Graciela and I can take a look at this and in 2021 start consulting with the regional office and the people to see how can we incorporate any of this into the decision-making process that we have. Regarding emergency action, in the federal government, emergency action is when the plane crashes. That's what I was told in Washington at a meeting when we requested for an emergency action, meaning that probably we won't have time to have an emergency action by February 28, 2021, because emergency action are done by the action agency, in this case, National Fisheries Service. But the important part here, Mr. Chairman, is to take this information and keep consulting with Dr. Shatter. And Dr. Shatter, by the way, is a member of the SSC, and we should take this information to continue the process that we have. By the way, Dr. Shatter is also helping us in the international level with the protection of spawning aggregation of species that use this strategy in their life history of each one of those species. So it is important we consider this information and add any actions in 2021-22 for the protection of the species. And just remember that any action that we have has to be in consideration with the socioeconomy of the area. So all that will be in play by the time that we put something together to address this issue.
Yeah, I understand that. I just would like to hear or give you opportunity to the council members to express themselves. And I have a question on the slide.
Marcos, go ahead and ask your question, and maybe that will entice some discussion because it's 12 o'clock already.
Okay. Can you go back that one? That slide. For me, it's very interesting because on the east coast of Puerto Rico, different than the west coast, the fishing grounds and the way the people behave in terms of fishing, the commercial fishermen on the east coast, they don't target the specific red hind like other areas of Puerto Rico, and more than that, there is not a specific area that is protected or is identified to go for the red hind like the west coast. Maybe that's the reason why we have that ratio different than the west. That's a possibility, Michelle. I'm reading this right, or can you make a comment on that?
Absolutely. I think that is part of the reason I think that there's this different sex ratio, but also the fishing methods used in the east versus the west from which these samples were collected is also different. So most of the east coast red hind came from traps, and most of the West Coast came from spearfishing. So that may also be a reason why there's this difference, but the most concerning one is what's happening at the aggregation site.
Vanessa?
Thank you, Marcos. Just to make a small comment, it's also, as you say, in the West, we have a lot of divers, so 300 divers. And this species is really well known in the area, and many people look for them. So all the fish markets pay practically three or $3.50 per pound. Divers are looking for it also. Thanks.
Okay. As a chairman, I would really like to have a discussion on this, on the future, and to explore which way we can address and learn about this new information, like Miguel said. And we're gonna get this information and see what it can be done, or which way we can accommodate a deeper discussion on this issue. Thank you very much for your presentation, Michelle.
I had a question, Marcus. My hand was up in the chat.
Go ahead. I'm sorry, Ed. Go ahead.
Okay. Excellent information. This is why it's so important to go into these closed areas for reevaluation. Now that this study has been done, are you saying that the larger fish remain even after the closure is there? So with the emergency closure that you're proposing, you want to say that you want to extend the closed area longer?
Michelle?
Yes, part of the recommendation we have made previously for the west coast of Puerto Rico in the EEZ was to shift, not extend, the closed season to be able to protect that last peak in the aggregation. So instead of 1 December to 28 February, we have proposed in the past 15 December to 15 March.
Thank you, Michelle.
So therefore, okay, it makes sense now. You're not extending it, you're just moving the peak time of the closed areas, because now you know exactly when the bigger fish are there and they're doing their stuff.
Michelle?
Right?
Correct.
Yeah, okay, thank you. Excellent information and presentation.
Marcos,
We
you have Richard and Vanessa.
Richard?
Yeah, I just wanted to reemphasize why this is really an issue now. And it's because, of course, that the lunar cycles do not follow the solar calendar. And while our initial decade of monitoring showed this problem to occur periodically, that's just how the moon happened to fall during that decade of work. And the decade coming up is going to be one where this is going to be a problem. She said that will occur 80% of the time. So it's really trying to point out this is something that we need to address soon, because it's going to be with us for a while. The only other solution would be to change to the Mayan lunar calendar, and that would solve all our problems because everything would be aligned. But then again, the world would have ended in 2012, so there's a downside. Thank you.
Vanessa?
Yes, Marcos, thank you. I just want to tell Michelle that I totally agree with the, we don't need to exchange the evidence that the calendar have been moved by the lunar cycle. So, and this is not only the red hind, the only species. There's also another species that are doing the same for the last 2 years. So we need to work on this, and for the help of the fisheries, and to instruct the fishermen also of the importance to maintain the calendar update. Thanks.
Graciela?
So I just have one question regarding enforcement. So the seasonal closure for red hind extends from the shoreline to the 200 nautical miles. Do we have any information on the success of enforcement during that time, the year that we have now, and how that might have contributed to the changes that we see in the population? And the question probably will bounce back to the enforcement officers and to the commercial fisheries.
Your question is directed to whom?
Well, if Michelle has the answer, it would be great, but if not, to the enforcement agents and to the local government and to the commercial fishers.
Michelle?
Yeah, I don't have any information on actual interventions, but when we've been sampling during the closed season, we have seen boats actively fishing for red hind at the aggregation site.
Anybody else have information about enforcement on this issue?
I know Yamitza Rodriguez on the line, if she can share some information, because I know she monitors the interventions. Right now, we don't have that type of information here. I would ask the fellows, colleagues within the NER after the meeting. I can provide information later on.
Marcos, the important part is not the enforcement at this time. It's just the presentation that Dr. Scherer is bringing to the attention of the group, which is a new information regarding the science, and also a request for changing the range of the closure that we have here to make sure that we cover the peak spawning time for the species.
Yes, Miguel. I would like to ask, I see the interest from Vanessa, from Eddie, from a few of the council members recognizing this important information. Which is the question is to Jocelyn, which is the best way we can address this as quick as possible, the discussion? Thank you, Jocelyn.
Thank you, Marcos. This is Jocelyn. One of the things I just wanted to circle back to that Graciela had said was just about the scope of the closures. So the federal regulations, we're talking about closed areas in federal waters, in particular time periods. I can't speak to any of the closures in the territorial waters, but we have on the books some closed areas and closed seasons to account for red hind spawning. And then I think the question is, do we need to adjust those time periods to account for different information? So we could look into the process for doing that, putting forward an amendment to revise some of the closed seasons or the closed areas to account for that information. So that's something that the council could request staff to look into. In terms of an emergency rule, I'd have to do some just additional looking at the scope of that, but I think Miguel mentioned that there are very narrow circumstances where we could have an emergency. So it doesn't initially seem like that would be appropriate, but it would be appropriate to reevaluate the management measures on the books to make sure that they're matching the best scientific information that we have.
I guess that's the best route, and we need guidance from you guys to make sure we follow science. Yamitza Rodriguez is requesting to speak.
Yes, hi. Regarding what Damaris mentioned, we can provide information regarding the interventions that the rangers have done with red hind. What we do is most of the times that they intervene in the water, the rangers come to the fisheries lab and we do the certification and if measurements are required of the fish for the law purposes or anything. And we have all that information since, I think since 2005 to 2019, it is mostly up to date. And we can give you information regarding how many interventions of red hind have been done during the seasonal closure. One thing to keep in mind is that most of the interventions that we received are from the West Coast. And since 2010, when the fishing regulations was amended, the seasonal closure applies island-wide. So we can try to collect if there are other interventions of red hind in the seasonal closure for other parts of the island, and we can provide how many interventions that the rangers have done in this period.
Thank you. Once you have that document, can you please send to the council and we, for us to have it and to distribute and,
sure. As
and
needed.
to have. Thank you very much, Jimmy.
Okay.
And we have Graciela.
Yes, please. So Michelle, if it's abrir la sierra only, that's completely within federal waters. So that's one thing. So would you suggest to begin with something like that, or would be better to look at the complete seasonal closure, and so that the government of Puerto Rico and the federal government move to December, from December 1st to December 15th to March 15th, the seasonal closure for everywhere?
Marcos?
Yes, Miguel?
If I may, we're only gonna solve this here. So now you received the proposal from Dr. Scherer. So I suggest that we allow the staff to meet with Ciro, Jocelyn, the local government, and Dr. Michelle Scherer, and then come to you with something that could be worked with.
I think it'll be more productive. Thank you, Miguel. Yeah. And we can move, we can do that. Just the last question that Graciela asked to Michelle, and we close the discussion.
Yeah, it's very simple. We have monitoring stations in other sites that are in federal waters, and we're seeing the same pattern. We do not have localized red hind sites in Puerto Rico jurisdictional waters yet that we could monitor and see if the same thing is happening in there.
Thank you, Michelle. There is anybody else for public comment?
You have Tony waiting.
Tony.
Yes, I'm sorry, Tony. Yeah, go ahead.
Yeah, I just like to touch base on something that really kind of bothered me today about this meeting. It was almost like we was trying to rush through an agenda. I think the lack of time for questioning, the trying to keep up with a time period to keep the meeting within, and I could understand that to a certain degree. It's almost like we was running a race. And I'm pretty sure there are other members that was out there that share the same thoughts that I share. And something that has nothing to do with this is Mr. McGrath was trying to log on to the meeting. I don't know how he got kicked off. And he told me he could not get back on the meeting for whatever the reason was. He was trying to log on and he just couldn't get back on. So I just thought I'd bring that to your attention. I don't know if we were running short on time. We had too much of a loaded agenda to deal with for the time frame. But I think we need to do a better job the next time.
Thank you for your input, Tony. I agree with you that it was a tight agenda. I share your opinion, and we're gonna keep working to make the meetings the best we can with the best information we can. Thank you very much for your input. We don't have anybody else. We are ready to adjourn the meeting.
One moment, Mr. Chair. We do have the CEDAR 80 Point Triggerfish appointment. So we've requested from the local governments their input in terms of the appointees, people who are experts on Point Trigger commercial or recreational fishers. So we are expecting their prompt response, and we have already confirmed the participation of Virginia and Jesus for the CEDAR 80 and collaboration with the Science Center and the CEDAR group to provide us with a successful assessment. So thank you. So you'll be appointing the CFMC appointees very soon.
Thank you, Graciela. I'm not missing anything else?
And I think Miguel, he's just dropped the signal, so I don't know if he's on.
Thank you for. Go ahead,
Chair.
Miguel.
This is John, not Miguel.
Okay, I'm sorry, John. Go ahead.
Well, one thing I just want to commend the science that we've seen today, and it's been fascinating. I really think that a lot of great work is going on. One thing that might be useful is if this science has not gone through the SSC, that to bring it to the SSC, because that's where there's time for some more evaluation of it from the scientific perspective. And I think that could help the council then get that science distilled into some concrete management actions. And it seems like that process might be an effective way to make sure that this science gets to become actionable on a management level. Thanks.
Yes, great input. Thank you very much. And I'm going to work out directly with the chairman of the SSC to make sure this information can be presented or arranged to inform the SSC. Thank you very much. I don't think we have anything else. Anybody else on the list? Thank you for your patience. Thank you for participating, Miguel.
Marco?
Yes.
Can you hear me?
Yes.
Sorry, yeah, because Liberty decided to drop the signal, so I'm in another computer. But I just want to thank everybody, and actually I'm taking note of Tony's concern. And certainly some people have problems in and out of the meeting, so they have to, because of problems in their section of where they are. So we are constantly admitting people to back in the meeting, and they send me, you know, the reason why they are in and out of the meeting. The last thing I was going to say is just this is our last meeting of the year. Hopefully we will not have another year like this, only we have COVID 2020 next year. But I want to thank everybody for their participation and patience during all this interesting time. All council members, all the chairs of our committees, and Habitat Advisory all the panels that we have. But I want to mention the ladies who were with us through the year. Thanks to them, the council have been able to continue working during all this time. And I just wanted to mention for the record, in the order that they sit at the council office, Angie, Graciela, Luz, Natalia, Iris, Diana, Liahai, and Christina. These ladies have done more than they were supposed to do on their position description. And they're also always willing and able to help us. So like this meeting, for example, we have the teamwork with Liahai, Natalia, Graciela, everybody involved. So for that, we are very grateful. And I wish you all happy holidays and a better 2021. And we'll see you guys in 2021. Thank you, Mr. Chairman.
Mr. Chairman, Manny wants to make an announcement.
Go ahead, Manny.
Yes, thank you, Marcos. I wanted to share with the council that our office selected the new enforcement officer. During the last meeting, I had briefed that we were working through the hiring process. And so we have selected an officer for USVI, Mr. Alex Terrero, and he's currently at the Federal Law Enforcement Training Center completing his basic training. Alex comes to us with a great deal of experience. He's got 13 years working with the Florida Fish and Wildlife Conservation Commission. And we're very happy to have him on board. My hope is maybe during the next meeting, we could do an introduction and have him speak with the council. And then the other thing I wanted to share was Matt Walia. He was selected as OLE's new compliance and council liaison. So Matt will be working directly with industry and the councils to address any concerns that may come up. Matt is he's participating on this call as well. And Matt, I'm not sure if do you want to anything you want to share to the group?
Yeah, I can just introduce myself. So I'm here and available to help as needed. So if there are any law enforcement concerns, you talked about some of the red hind and the EZ or closure areas, please direct them my way or Manny's way, and we'll do what we can to help address that. There's great presentations by the outreach liaisons. I plan on reaching contact with you guys as well and look forward to in the future, we can work with you at more island to island level. So that's all. Look forward to working with you guys. Thank you.
Thank you.
Thank you very much. And thank you to all. Merry Christmas. Happy New Year. I hope everybody stays safe and healthy. And thank you for your support. And now we are ready to adjourn the meeting. Thank you very much.
Gracias a todos y a todas. Se cuidan. Y felicidades.
Happy holidays, everybody.
Un abrazo.
Cuídense mucho.
Hasta luego.
Hasta luego a todos. Bye.
Bye.